across Africa and beyond. He's also deeply engaged in advancing the global understanding of antimicrobial resistance, population genetics, and One Health approaches, linking human environment and microbial ecosystems. In addition, he also contributes to the scientific community as a community reviewer and an editorial board member of the Evolutionary Bioinformatics section of Frontiers in Bioinformatics Journal. So in today's presentation, Dr. Kuja will discuss how to unlock genetic diversity for sustainable development of Africa. So ladies and gentlemen, please join me in warmly welcoming Dr. Josiah Kuja.
Thanks, Samuel, for the good introduction. And as you said, I'm Josiah Kuja. And the invitation to speak here was on the African Biogenome Project. But since, as you said, I'm a microbiologist and a genomics expert, the microbiologist in the house will not feel good if I go without talking about microbiology and AMR. So 5% of my presentation will be on AMR, while 95% will be on African Biogenome Project. Something? Not really. Oh, okay. So the outline for the presentation, I'll just talk about on the concept or conceptualization of Africa BP, and also talk on the Africa BP itself, and also the importance of genomic research in Africa, as well as how we really drive the African Biogenome Project through collaboration and partnerships, and also the milestone so far for the last 5 years since Africa BP was placed or launched. So Africa BP, the concept came up in 2019, and the COVID outbreak just really made it become a success because we were closed indoors and we were able to go virtual and just find something important to do. So most of the scientists or scholars who really met to establish Africa BP had enough time to spend and talk more on how to go about it. And in 2021, it was launched. And 2022, we managed to publish a position paper in Nature, just giving a roadmap of what Africa BP plans to do, and also the mission of Africa BP. So why Africa? Why are we talking about Africa Biogenome Project? So Africa hosts extraordinary biodiversity, cutting from the south to the northern Africa, East African, Central Africa, and also West Africa. However, the African species remain underrepresented in the global genomic database. This means that if you want to do reference to the African species, you can not easily get this species for reference for local studies and also for research. However, and again, most of these species are exposed to climate change, habitat loss, emerging pathogens, and ecosystem disruptions that are making them to be really exposed to extinction. So Africa must lead genomic discovery, infrastructure, analysis, and policy translation to really conserve and sustain our ecosystems. So the intention is for Africa to leverage on indigenous biodiversity, including plants, animals, which have got numerous advantages. The traits for some of the species within the African biodiversity have got resilience to disease and pest resistance, drought tolerance, and also stress resistance. But in case they're exposed to extinction, then some of these genomic characteristics or strength will be lost. There is a need for the plants and animals to be fully characterized. So, with all those statements, so what? Because we are racing against time. We can wait with the climate change, and then we know the consequences, or we can do something as scholars within the continent in collaboration with the experts globally to really save the situation. And the latter will have more benefits for us and the future generations. So, what's Africa BP, the African Biogenome Project, abbreviated as Africa BP? It is a large-scale Pan-African initiative aimed at sequencing over 100 species in the next 10 years. So by 2021, when it was being launched, we talked of within the next 10 years, and we continue saying within the next 10 years, because we are really working hard to achieve this within the next 10 years. It's a continental effort led by scholars in the continent and global. It's also led with the African institutions, the universities, research institutions, with the like-minded, to support the infrastructure, including the computational abilities. What are the goals of the Africa BP? So the first goal is to act as a forum for the non-human space. And this will help to coordinate the sequencing of the non-human genome. These will include the aquatic, terrestrial, and even in collaboration with the medical initiatives like the African CDC Pathogen Genomic Initiative, just to inform on the One Health initiative. How can we work based on the One Health approach while focusing on the non-human space? So we aim to initiate, develop, and support non-human genome sequencing efforts. And as I said initially, we are aiming at sequencing over 100,000 species in Africa. And this one requires a lot of energy, support, and also a goodwill to achieve this. And this sequencing these species will inform the genomic or genome availability in the global database for reference for our local studies, for our local students, and also for the species identification. We also need, another goal is to deploy genomic data to inform policy and build capacity in Africa. So the sequencing data will contribute to regional biobanks, including the Africa CDC Pathogen Genomic Initiative and also the Africa Union Inter-African Bureau for Animal Resources. What are the importance of genomics that Africa BP is really working towards? Because the main tool that Africa BP rides on is the genomics. So the importance of genomics in research in Africa. So we really aim at conservation, sequencing. Priority focuses on the endemic species, and more so those which are threatened within their respective ecosystems. And the diverse and unique ecosystems are threatened by the climate change, habitat destruction, illegal trade of animals and plants, pollution, overgrazing and agricultural expansion. And all these features, we really understand them as biologists. So genomics is now a fundamental infrastructure, and I believe all of us, we know this is really important for the next steps. What can genomics do that Africa BP is really championing and riding on it? It will enable data-driven solutions, create an understanding of climate change. If you have the resilient organisms, are they endangered or are they able to exist and inform future sustainability? It impacts on the adaptation of species. And again, it enables pathogen detection and inform policy decisions. And for the microbiologist, I will focus a little bit on the pathogen detection. I have a slide for the pathogen detection, and that's also part of my work that I do now. And it will also tell or inform how genomics is being used really within the Africa BP and the entire space for genomics and pathogen detection to inform policy decisions. So this is just a phylogenetic tree, or for phylogenomics. So for microbiologists, you are at home, and I'll explain a bit of what is the relation there. So genomics and pathogen evolution, and this is in the case of the ESBL gene landscape between the hospitals and communities. So what can genomics tell us? So this figure just shows like samples were taken from hospitals and communities. And again, we have now the classification of the samples. Mostly these are stool samples. Then you do culturing, then whole genome sequencing. Then you have the downstream genomic analysis, just to really interrogate the sequence or the genomic content and come up with the genomic elements that can be used to inform the gene, ESBL gene landscape or antimicrobial resistant landscape. And then how can this be used really to inform public health systems for management? So this phylogenomics tree shows like samples are from the communities, and here we have got the ancestry, the sources of these, or the families, the sequence families. So we have various families that they came from. And this tree have got two major clusters. But these clusters are regardless of their sources, simply showing that the antimicrobial resistance within the hospitals is expanding. It's a bidirectional expansion and transmission between the hospitals and the community. And that makes it so difficult, really, to understand control measures and even the IPC approaches. Okay? So then what do you do? But you have to understand what are the ancestry of these isolates. Then, because you know the genes are transmitted sufficiently through the plasmids, because we have the horizontal gene transfer through plasmids, which is really fueling antimicrobial resistance. And as well, we can have the clonal expansion, which is really facilitated through the chromosome. But from these samples, we see that chromosomal expansion is only 30%, while 70% is through the plasmid. It means that within the health space and the community setting, there's a lot of interaction of organisms. So we are able to tell which gene is really fueling antimicrobial resistance. We have the CTXM15 and also CTXM27 and a few of the 55 really fueling antimicrobial resistance within the hospitals and also the community. And also we have some of the clades. The clades are just the ancestry of these isolates, really supporting through their variation over time, diverging and changing into new variants. So how do we understand the location? Because when we talk of plasmids, then how are they really occurring? What make these organisms be transmitted widely through the plasmids? Because you know that resistance is successful due to virulence and also containing the resistant genes. So further, you can use the genomics to tell what is contained within what. So here we have the plasmids, the incompatibility family plasmids, and these are the antimicrobial resistant genes. We can see here STXN15 is widely represented, and it is contained also within the plasmids. But these plasmids also contain the virulence genes. These are just the classes of virulence genes. So the plasmids here contain both the virulence genes and the resistant genes. And this is what is really fueling antimicrobial resistance within the hospitals and community settings. So microbiologists, feel at home. We go back to African Biogenome Project. So how is the African Biogenome Project run, and what is really making it become a continental initiative is through collaboration and partnership. So you need strength, both within the north and the southern collaborations. It means we have got the local collaborations and also the international collaborations, just to make create an environment for inclusivity in genomic research. Again, this will help us. This has also helped us build African to African partnerships through institutions, and also leveraging on existing infrastructure. So if University of Eldoret has got a sequencer, I do not need to buy a sequencer, but I need to buy another machine that will really complement the process of really sequencing within the continent. And also, it also enables us to learn from the best practices, because we are of different expertise. I'm a bioinformatician, then Samuel is a biologist, molecular biologist. We need all those expertise work together and leverage on also the best practices. So this is the outlook of Partnership Africa BP works with partners, institutional partners, corporates. So institutions are the universities, research institutions. Then you have the corporates. So the corporates are the business institutions like the INCABA, who really assist in sequencing, because when they have a new kit which they have to test, they just call Africa BP, like, can you get this number of samples to sequence and for just like a pioneer exercise for sequencing to see how it really works. Also, you have the projects. So other partners are the project associated partners. So in this case, if somebody has got a project which is running, maybe within the SMBE, then can recently collaborate with Africa BP to leverage on the reagents that are needed, and also to add to the number of sequences or number of genomes that are being sequenced. But we have leading high number of African institutions that are really in partnership. And as it now, we have got over 60 partners who are really helping us run Africa BP. So the goal is to sequence 95% of African BP genomes within the continent in collaboration with the global institutions. And this will generate really huge data. And the aim is to analyze this data within the continent with the support of young scientists and in supervision with the senior scientist. This may take long. As I said at the introduction, that we are aiming to do this within 10 years. It may take long. And these gaps, if these gaps are not really addressed. So we are really focusing on the infrastructural gaps that must be addressed within the continent, and also genomics and bioinformatics capacity, which are really championed by the Africa BP leadership. So to address these issues, in the previous slide, we launched the Africa BP Open Institute for Genomics and Bioinformatics. So it is an open institute. It means that you can be taught, or we can be taught, or collaborate and share information at your convenience. So it's open institute whereby if you're in Eldoret, I'm in Nairobi, or in the US, then you can easily talk and chat way forward on how to address these issues. So the main aim of Open Institute is to develop a curriculum that will factor in students from the undergraduate up to the postgraduate level. Also to promote the use of technology within the African institutions, and also promote grassroots, take genomics to the grassroots, okay? Because when you just hear about ILRI doing sequencing, what is sequencing, but taking genomics to the grassroots. So we understand what is genomics and what is the impact of genomics within the continent. Then again, to give a room for the corporates, we give a room for the sequencing companies to come and collaborate with the scientists to really know how we can better have leverage on sequencing through collaboration with the partners, the corporates who come and talk to students, talk to researchers on how we can best use genomics to leverage on sequencing and genomic applications. Then also data ownership. So this year we are talking about digital data, and data ownership and genomics. And that's part of the Open Institute, what is taught and what is really exercised. So, Africa BP has also been championing development or to develop early career scientists. They need to capacity build early career scientists. So we need skills because, you know, including myself, at some point I had to step out of Africa, then come back, okay? But while within the continent, what do I really do? Do I focus on my work and forget about the young students who really need supervision? So we need skills, focusing on the taxonomy, genomics, bioinformatics, and also computer science, really to drive this initiative. Also, we need to retain, by retaining these African talents, so we must work hard. James, we must really work hard to retain these young scientists within the country, but in collaboration with the international space, global space, to make them have the optimal skills in the applications. Thus, the need to build genomics and bioinformatics infrastructure in Africa to harness skills for the country and the regions. Also develop homegrown research projects. So, what are the milestones with all those, what Africa BP has been doing since it was launched in 2021? So we have been having regional workshops. So Africa BP is divided into 4 major nodes. We have the North Africa node for the North African countries. We have got the West Africa node for the West African countries. We have got the East and Central Africa node for East African and the Central African countries. That's the Republic of African, Congo, and also Cameroon. And also we have the South African node for the South African countries. And we have been able to run 3 successive regional workshops. This is basically focusing on genomics and bioinformatics and involving university students, scientists, and laboratory-based expertise. And this started in 2023 all the way to 2025. But as we speak today, the West African have already taken part in the 4th regional workshop. So this usually occurs with 2 days of conference-style presentations, followed by 3 days of hands-on genomics exercises or practicals. We have the corporates, the genomic corporates, the genomic corporates communities come and train students on hands-on. So if it's long-read sequencing, short-read sequencing, then they train students on these skills. Then Africa BP creates space for that in collaboration with the corporates. So in 2025, you have North Africa doing their regional training in October. Then you got the South Africa did theirs in August 2025, West East, and Central Africa had one in July. And this year, we already have West African that took place just in the last week, actually, for the 2026. So training over, this now a good show for what still takes place after the in-house or the 2 days conference with experts, with panel discussions. Then you have got the 3 days of hands-on training. So like in this case, you have got the students being trained by the INCABA Biotech East Africa. And also you have separations from South African, taking the student through, or participant through the hands-on genomic genomic extraction and moving forward to sequencing. And this usually takes 3 days. Another milestone for the regional workshop is that this is an indication for 2025, where we have in East Africa attendance was over 1,000, and in North Africa it was at 978 participation, and South Africa was at 835 participants, and West Africa had, it's indicating 40, but we have got a lot of intersections of occurrence, but generally we have got over 500 participation going forward for training within the continent. So in addition to the initial publication, we also have a roadmap on how to take genomics and bioinformatics to the grassroots by establishing African genomics and bioinformatics programs to the institutions and collaborators, as well as the partners. So we have also, in collaboration with the international partners, we have also been able to train leaders, because I am here today, but what of tomorrow? So what we do is to capacity build young scientists on genomics and bioinformatics. So the international collaborators for the institutional levels have been able, like we have the Karlsruhe Institute for Genomics Biology has been able to train 5 genomic leaders, African genomic leaders, and these are postgraduate students from the East Africa, five regions: East Africa, Central Africa, North Africa, West Africa, and South Africa. And these candidates, now they are leaders of specific groups within the African Biogenome Project. So whatever we learn through our collaboration is being implemented locally. Regarding training and the milestones, there's an upcoming training for the East and Central African node, and this will happen in July, August at Mount Kenya University. But this will be expanded more for registration. So while talking over the milestone and what Africa BP has been doing, we have also got challenges. It has not been really smooth, having all these partners coming on board and also moving from institutions to really inform, like, this is our mission, this is our vision, and we really want to put our content in this status. So the challenges and the representation of the African genomes, that was the introduction part of it. And also fragmented infrastructure, because when we started, the focus was on South Africa, having the sequencers and could easily support. So by then, even the INCABA was able to support long-read sequencing, and we have got voucher samples which are really sequenced by then. But now there's some development, and we are working on a a statistics report which will really show what's the status of African genomics applications compared at the initial state of Africa BP in 2022 and the current status, because now even in West Africa there are long-read sequencers which have been installed by the INCABA, and some in East Africa also coming up. And North Africa, at the moment, V5 University, there's development of the genomic institution that is focusing on sequencing. So a lot has been growing, and they're supporting Africa BP initiative as well. Then also you have the limited computational capacity, not only having computers, but do you have computational scientists who can really easily help or really help in the development of the needful for success of Africa BP or success for the genomic and sequencing on Africa. Then we have got inequitable collaborations. So even up to date, some collaborators, when come to Africa BP looking for a unidirectional support, but look for bidirectional support. You may not have an equipment. Africa BP will be having that equipment. Then, do you have the samples to sequence? Do you have the reagents to support the sequencing? Or do you have a student who has been supporting the Africa BP initiative and who really want to move to Europe for 6 months experience on genomics? Then also on bioinformatics and data protection. That's also still a challenge, but we're making progress on this. Then we may have some food for thought with all those taking place in Africa setting. So training of African early career scientists needs to go hand in hand with mechanisms of retraining them on the continent. So unfortunately, James is away, but can these young scientists be positioned in the universities and get some good support and pay to keep them busy within the region? Then again, having good infrastructure. Good infrastructure would just mean having these bioinformatics enabled computer for Linux applications, and then they're trained. Because I was trained in my bachelor's, I was trained genomics and bioinformatics by being told, get the sequences, deposit in NCBI, then you get your phylogenetic tree. But can we do that locally? As Josiah, myself, having this computer, can I able to share with the students and tell them this is the Linux platform and this is how to go about it? Then opportunities for promotions, growth, and development. So we need really to support the young minds, young scientists, really to be able to reach out to the information and capacity to grow our institutions. So the importance of role of Africa BP Open Institute regional workshops. So through the regional workshops, Open Institute for the regional workshops, we aim at bridging capacity, bridging the gaps among the young scientists in Africa by training the genomics and bioinformatics, and how they can easily apply this in their daily or routine project within the institutions. And also discussing capacity building and training needs. So I cannot just come to the University of Eldoret and start training the advanced bioinformatics, but I must understand what has Samuel done, what has James done, then where do you start from with my expertise? Then determining a common curricula that can be developed to address these needs. This is also very important on how to approach the implementation of Africa BP objectives. So we can move barriers by, so how we have been able to reach out by creating the Open Institute, it's just by, we create a platform where everyone else, regardless of your location, you are able to get access to the trainings and information. And if possible, you are able to train and have a physical presence. So this yet is at MKU in Thika. If you are you can ask the university to support your movement to MKU for hands-on training. Then publication as well. So some journals always try to waiver publication fees to the low and middle income country students. We can focus on this as well. Then access to conferences. Okay. So are there any student outside University of Eldoret who is present here? Yeah. And thanks to your mentors and supervisors for letting you reach and how they be exposed to such kind of a forum. Because this will open up your network and you understand like who is who and how can you grow by working hard in biology. Science is not easy, but through this platform, you'll be able to go up the ladder. And also determine common curricula that can be developed to address the needs, just as in the previous. So this will really facilitate the process successfully. So the future of African genomics will not be defined solely by how many genomes we sequence, whether there are 100,000, but by whether Africa builds the scientific capacity, infrastructure, and leadership required to transform genomic knowledge into sustainable development, health resilience, and biodiversity stewardship. It doesn't matter how much you sequence, because people have been sequencing, scientists have been sequencing, but what are the policy implementation process? Are they really available in the databases? So that's what we're really working on to ensure that there's a sustainable way of having this data available for decision making and policy information. Again, if University of Eldoret, I'm the one in charge of partnership and collaboration framework for the Africa BP. University of Eldoret is not a partner to Africa BP. So, Sam, if you want to collaborate and partner with Africa BP, this is the link. Just go to that website, then do the necessary, like your information, email, and then someone will get back to you. Then you get an invitation to be a partner to Africa BP. Thank you very much for listening. And here is the QR code for this year's East and Central African Africa BP Open Institute Regional Training. And thanks to SMBE for the invitation. Thank you.
Thank you, Dr. Kuja, for a wonderful presentation and showing us how Africa is taking leadership to generate information for ourselves that we can consume, obviously, to improve our situation. I must say that I'm one of the ILRI capacity development workshop within Kaba Training as on the Oxford Nanopore. So, yeah, it's interesting to see how those skills can be used to relate problems. So we open the forum for discussions, comments, questions. So the floor is open.
Yeah, thank you. Thank you very much for this really interesting presentation, and it's a fascinating effort. My question, though, is there's one aspect which is perhaps central, which was not so much mentioned in your talk, and that is money. And this is all very costly, and money comes often with interdependencies or interests. So I wonder how you address that issue. I mean, it reminds me a bit of, you may know, perhaps some years ago we had these offers from BGI and Beijing sequencing everything for free. And I said, why do they give that for free? There should be some interest. What is the real interest they have? And I cannot say that I resolved it, but I wonder, how is it here? How can you ensure that it stays really pure African if the money may come from elsewhere?
Yeah, thanks for that. And yeah, if you really noted through the presentation, I was talking of collaboration and also with partnership. So initial sequencing effort was really supported by INCAB in collaboration with Illumina, just to enable all genome long-read sequencing, which was a bit of a success. So we have really been working closely with the partners to enable us do the sequencing. And again, training, which is really expensive, just getting student or 5 students from South Africa all the way to North Africa to travel to the US for training for 6 months is quite expensive. But the partner institutions are able to support this. So that's the level of collaboration. We really we are not really a profit-based initiative. This is a consortium that's based on the scholarship, goodwill, understanding of the subject. So the partners have been playing a very important role in this. And we are working as well on grants just to support the local trainings. And we are looking forward to whereby this will now be channeled to the regions, the North support. So we've done grants to Wellcome Trust, we've done grants to Illumina, Beta Initiative, just hoping that all will be in place. And that's where collaboration comes needful with the partners globally. We cannot be alone. We need the global support. Yeah.
Yes, thank you for the very informative presentation. So in South Africa, we have the 1K SA project from Diplomics. So in the spirit of collaboration, is that part of the 100,000 K project, or not part of it, but is the linkage between the two projects? And then secondly, just, and this is probably very difficult to answer, in terms of the 100K aim, where exactly do we have a sense of where we stand at this point in terms of the number of sequenced genomes across all platforms and institutions?
I'll start with the last question because the first one is a bit easier, since Diplomics is a partner to Africa BP. So regarding the capability really to manage the 100,000 species sequencing, it's difficult, to be honest, but still through the collaboration and the partners' ability, we see this to come through, and even establishing the servers for retention and also analysis interfaces, we believe it will work. Because now, in 2022, when we are starting or really getting engaged into Africa BP, we did not have we didn't have a long-read sequencing in West Africa. But through the partnership and also with the collaboration, Illumina saw it wise to really expand the long-read sequencing platform from South African to West African through Inkaba Biotech. And even this, it was last 2 years, the Mohammed V5 University in Morocco is establishing a genomic center, which will really boost the Africa BP vision and mission. So we are hopeful, and with their support as well. So the first question, Diplomics is a partner to Africa BP, and 1K project is able to come in as a project associate project partner. Yes, and I can advise, in case you need any information, just go through the website, or you can just email me direct, then I'll redirect the need for partnership. Yeah, welcome.
Yeah, thank you very much for your nice talk and the really nice initiative of Africa BP. My question is a bit related to the first question about funding. It's a really a big issue, and you mentioned that your collaborators in Kwaba and Illumina, they are helping you for sequencing all those stats. That's quite good, but I'm still interested in knowing, like, for example, for you, I don't know, other leaders, maybe they have salary or something. Where is that money coming from, the major source of funding? And also for universities where you have already organized some workshop, I believe when you gather people, at least you have to give them water. So where did you get those money from to, something like that? Or the universities, for example, in West Africa or South Africa, providing for this kind of thing? And my second question is related to this, what Prof has already said. It's about servers for storing data. So where your server's capacity, or I don't know, something like that, where do you store your data? And my last question is about crop pathogens. So far, have you collected or sequenced any parasite or pathogens that affect crops? Thank you.
Okay, thank you. Regarding the pathogens, because I'm a microbiologist, and I'll talk on that. For now, there are no specific crop pest-based pathogens that are being sequenced, because our focus generally is to do long-read sequencing, and the project partners, with their institutional ethics and also with the sample retention and provision, it has to go the normal process with the sample sharing or species sharing, and also with the sequence. So as of now, we don't have any specific crop pest-based pathogen that has been sequenced, but we have over 1,000 sequences that have been done on plants. There are some aquatic animals that have been done. And for the surveys right now, we have been working with a server from the University of South Africa, which has been supporting even the bioinformatics work and also the annotation process. And also in collaboration with we have got, it's called VGP, the Vertebrate Genome Project, also has been really supporting this process through the Galaxy platform for analysis. Regarding the money, just as I have said, the partners have been so wonderful. And yes, myself, because Africa BP is a consortium, and the leadership, they're employed, some are getting their money. But once you have got these activities, we have the goodwill ambassadors and the corporate institution, the business units or the groups, they are really willing to support, because we meet them, tell them this is our mission, this is our intention as Africa BP, to teach and elevate young scientists, and they say, yes, we can come in and support that for the water, for the lunch, and all those. And it has been happening very successfully.
Yeah, just a small follow-up question. And have you been also getting support from African Union?
Yeah, we haven't really got the financial, but we have got the goodwill support, because once we even need somebody just say this is taking place, then you can do a follow-up. And even this through the Inter-African Bureau for Animal Resources, they have the goodwill to really push forward the Africa BP initiative.
Yeah, I wanted to understand whether there is something like in other projects there that have been also available for genome sequencing. People could submit suggestions for species to be sequenced. And I think that can be very helpful, because maybe someone like me in South Africa doesn't know any person that has similar interests in West Africa. So through such things, the consortium could also start to connect people with each other. Yeah? So you're talking about partnerships, but the partnerships sometimes need also to be initiated in a way. So with such things of maybe submitting suggestions and also explaining what type of resources you have and what you can bring in, there would be also a chance then to connect people with each other that have similar interests, so that they can join forces, maybe for joint proposals and stuff like that. Is there some idea like this?
Absolutely, yes. And that one you come in as an associate project partner, where you have got a project or a species or samples to sequence. You come and say, this is what I have, I've got this kind of sample to sequence. And yes, we've been doing that. That's where we started from in 2022, where we could just called sample submission. Then they were to be to South Africa for sequencing. So it's really possible. And also within the Africa BP, create a platform for scientists to interact. South African will be able to interact with the West African, North African, East African, and even European settings. So there's a platform for that level of interaction once you become partners within Africa BP. Yeah. So please feel free to go ahead and reach out to Africa BP. You'll get me on the spot and I'll just redirect the platform for discussions.
It seems attracting a lot of reactions.
Hello, thank you for your presentation and also for the project. I just wanted to ask, is it also one of your goals to create like a data bank for these sequences? And if so, do this data bank only include sequences that are generated in the project? Because there's also other projects generating genomes, right, of pathogens, of biodiversity. Will this also be included? Is there like a research phase? Yeah.
Absolutely. Of course, we cannot achieve this without biobanks, and we cannot achieve it without the biobanks. And indeed, there's a provision for that, for the biobanks. And this biobank, the data to be stored are the data that is African-based generated data. But we have got, because we have got like, there's one crop, one African, there's some projects going on, for example, but we collaborate with them because this data is already outside, but they are willing really to collaborate and continue sequencing, supporting African-based local sequencing, and then feeding in those data sets to the biobank. Yeah. So all this just come by meeting, then discussing, oh, you've been doing this somewhere else. Oh, okay. How can we move forward? By really expanding on the sequencing and also on the data management. So very possible.
Yeah. Thank you for your presentation and the sharing about the initiative, which is a good one. One question. Is there an effort also to initiate other multi-omics projects? Because we know, for instance, proteins could be really the main functional compounds to really be studied sometime, especially for diseases and such. So are there those projects also in mind with the projects you're having?
Yeah, that's a good question, because when I talk of institutional partners, project partners, and also you got the corporate partners. But you also have working groups within the Africa BP, sequencing, annotation, data analysis, digital and sequencing group. All these students who do not have projects, who do not have institutions, who do not have corporate capacity, they join Africa BP into the working groups. Why do we have working groups? The working groups are there for diverse expertise. For us to understand if an organism, or if a plant, or if dairy cattle are able to be resilient to pest or to climate change, we must understand the metabolomic content of this organism. And that's now the proteomics. So we need really to digest the specific organism to understand this, after sequencing, what is contained within the genome, what genes are really available. So all those nitty-gritties of genomics, proteomics are really welcome, because we want to communicate like, oh, okay, this animal from the northeastern has got these specific genes for climate or for drought resilience. And that we must go to the inside, dissect the sequences and know which type of genes are available and proteins really to understand the organism. So as students, you can join Africa BP into the working groups, and your expertise is really welcomed. Not only students, but if you don't have project, if you don't have institutional head like capacity, come as a working group, and yeah, you'll be accommodated.
Good morning. Sorry I came late, but I got a little bit of your talk, and I'm really interested to, first of all, I must say it's really relaxing to see people who have done this much of genomics in Africa. And there are challenges that we still face, like, for example, when analyzing all genome sequencing or all exome sequencing, we always feel that the African reference genome may not actually reflect the genomes that are available. I know there's a Yoruba genome, but again, North Africans are very different from East Africans. So I don't know, are there any initiatives to build genomes that could really answer some of these problems that we face when analyzing genomics data? Yeah, so that we don't get a lot of voices, instead get something that is close to really giving us the significance of each and every variant that we see.
That is a yes. So that's actually, in a nutshell, that's what Africa BP is really championing within the continent. Just do species level, all genome sequencing, long-read sequencing, and have that as a reference genome, aiming at 100,000 and over or so species. Yes, and this will be available for reference on the African species. Yeah. So we have got both the North African, South Africa, East Africa, Central African, West African scientists coming together, and I think it should be a partner to Africa BP, so that you get the help and also the platform to air such kind of needs. But they're already in the process of really making such kind of sequences available. Yeah.
Maybe in connection with that, because Dr. Torore has mentioned about the Yorubas, it seems the data is incomplete without the human genome side. Is there a specific reason why you didn't want to venture into that?
Yeah, we talk about non-human species sequencing, because also human sequencing is already covered within the health sector or the health interface, and we do not want to create unnecessary competition, because we want to focus also the health sector as collaborators, really to, for sure we need that complement, but we don't want to bring in the competition. So we want to give the health scientists a platform to really up the game, as we also work from the non-human species side.
Okay. Thank you for your presentation. My name is Daisy. I have a few questions, and you indicated that within 10 years, the goal is to sequence around 100,000 species. So what is the progress so far, given that it's almost 5 years since the inception of the project? And then two, is there any policy that so far has been informed from the findings or from the sequencing that has been done so far? And maybe my last question is, so you have indicated that there are regional trainings. So what next for the participants? What for the participants of the trainings from your end? Thank you.
So thanks. I'll start with the last one, which I can easily remember. And in my last slide, there was a QR code for the East and Central African regional workshop that is forthcoming in July at MKU, Mount Kenya University in Thika. And for the local students, is worth applying. I think deadlines are fast approaching. They're over here. So just try scanning this. It will redirect you for the regional trainings. And these regional trainings, if you want hands-on, you can attend the local one. But if you have support for travel, then you can even go for South African training, West African training. But still, you can register for virtual training in case you do not want really to go, or you are not able to go for the hands-on training. So if you scan this QR code, it will lead you to the Google form that you'll fill. Then you'll be in the selection platform. The deadlines are fast approaching, I think somewhere in June, 15th June. Yeah. On policy-informed sequencing-based approaches, yes, we have sequenced what has been cataloged, because we have to go through annotation, then cataloging, then you have now a way to communicate this. But we've done over 1,000 samples, if I remember well, because we have got nodes working differently. And the sequencers keep on coming. So we've got over 1,000 samples which have been sequenced. And once we sequence, we publish. There's a publication on the South African goat. And this publication shows like this is the first goat which has been sequenced in African whole genome sequencing, long-read sequencing, and it has got all the information of the content of this genome. And that's how we do the process. We get the sequences, then go to the nitty-gritties of the sequence genome, and give information on that specific species and what it contains. And that comes now to the expert-based applications. So this kind of organism, the policy information is that it tells whether it is, we can know, does it have the genes for resistance or climate resilience, drought resilience, pest resilience? If you get such kind of information, then this has to be communicated back to the relevant agencies within the government setting. Then regarding the 10 years goal, yes, we're working hard, and join us, help us push it forward.
Thank you. So is there a follow-up that is done after you've trained the early career? Is there some initiative to follow maybe what they are doing, or if they are also maybe pushing the ball forward with the training that they have received?
Absolutely, yes. Because if I can give an example of Inkaba Biotech, what they do is I remember the first cohort was on sequencing good practices, just like a class, like sequencing good practices. How do you set your platform if you want to start receiving a sequence? Okay. Then they went into the lab, wet lab, DNA extract. Then the last year's practical was on sequence management. Then I don't know what they are for this year. They'll tell us very soon for East Africa. Like, these are the state of data analysis and interpretation. So there's a follow-up. And since it's an open institute, we may have even the business people logging in. So wherever, science, our science is, whoever is, I've got that science drive will maintain and move up the ladder, up to the final state of understanding the genomics and bioinformatics needs. So there's that provision. Whoever wants to follow up science, because some will come and change their field. But whoever started from 2023, they're somewhere still, because I've got the database, just following like this is this, achieving this. And such kind of participants are the ones who are likely to be recommended for the exchange programs to the partners, global partners. Yeah, because when you apply, we have to interrogate, have you been participating? What are your interests? Or you just came once in 2023, now you want to go and benefit from the exchange now? Yeah.
Okay, so, yeah, thank you. Yeah, thank you, our guest. So my comment is on, thank you also for this project, but my request is that there is need to increase the training for bioinformaticians on the continent to support the analytics. Yeah, because currently what I know is that we have very few bioinformaticians. And because of that, they are obviously overstretched. It is very difficult to find one because they are always engaged on demand, on travel and all that. Yeah, in our university here, if we have, you would find that one is overstretched because he also has to teach, he also has to supervise, he also has to do research and other activities. So really there's need to train more bioinformaticians to support these fields. Yeah.
That's true, James, because I think yesterday there was a student presenting. Did Vivian present? Vivian? Okay. So Vivian is an example of a bioinformatician taking masters at JQuat, who is my student. I can't do it alone, so I must just capacity build someone with energy. And it's so difficult that you must have the passion to just go up. So we have Vivian, who is really soon defending, publishing her work and defending. Then we have got one called Paul Kagame, who is one of the committee, steering committee for the East African and Central African regional workshop coordinator. Is defending his thesis at the Ghent University in Belgium. That is mid of June. Then he comes back, also a student that has been trained too by informatics. So yes, it's possible. And myself, I'll be willing in case of, I don't have, I don't say I don't have time because I've got the will to really support. But in case, just inform me that a student by informatician or by informatics who is interested, I do the little I can just to ensure that somebody somewhere is really appreciating the initiative and support from my expertise.
So thank you, Dr. Kuja, for your interesting talk, and also for the audience for your engagements. So join me in appreciating Dr. Kuja and Sharon. I think lead us in a nice, hearty congratulations.
Thank you so much, Dr. Josiah, for a good topic, a good presentation, cutting all along the fields, inclusive of genomics and bioinformatics, as well as AMR and microbiology aspect of it. I think he deserves a good applause for that. Let's have good 5 factorials for him. As we say, it's a superb work. Maybe we try just 1, 2, 3, 4, 5, as we did yesterday. Can we start? 1, 2. Thank you so much.
Thank you. So this morning, before tea, we have 2 more presenters. The next presenter is Sharon. So Sharon, you can take the stage. Sharon will be presenting on identification of genes driving antibiotic resistance of gram-negative pathogenic bacteria from the water systems of Eldoret City. So it's interesting to see how Eldoret has received a city status. It's good to see how the water systems are doing.
Thank you so much, Chair, and everyone. Good morning. I'm Sharon Nauma from University of Eldoret. I'm here to enroll for my PhD studies. I'm also a graduate assistant at the Department of Chemistry and Biochemistry at the University of Eldoret. Today, I'm so privileged to present part of my work, part of my research work that I did under my master's level, which links genomics and evolution of antimicrobial resistance in agriculture, medicine, and environment. My topic is about identification of genes driving antibiotic resistance in the plasmids of gram-negative pathogenic bacteria that were isolated from Reverse Oceani and wastewater systems of Eldoret City in Kenya. Just to introduce my work, antibiotic resistance in pathogenic bacteria is a current and critical global threat that affect many public health sectors, whose spread resulted to 4.95 million of deaths in 2019, the reports that were confirmed by World Health Organization as well as other researchers. Rivers and wastewater systems receive a lot of influxes, including of the wastewater, that do not only carry bacteria and antibiotic residues, but along with other pollutants, such as heavy metals, microplastics, and all that, which really makes these bacteria to, with time, to develop some of the mechanism, enabling them to survive and adapt to the stress regarding chemicals that interact within the environment. These continued interaction of bacteria and antibiotic residues have resulted to a number of antibiotic-resistant genes, which are most acquired by the plasmids as they interact within the environment. And these plasmids are capable of spreading as well as transferring these antibiotic-resistant genes to various bacterial pathogens and other microbes within the environment through horizontal gene transfer mechanisms, such as the conjugation as well as transformation processes. Just the environmental view of what is happening. As we are told, there is much interaction between bacteria as well as other pollutants within the environment, especially in the aquatic sites, whereby rivers receive a number of discharges, or we can say the effluents from domestic areas, from cuticle, industrial areas, from the wastewater treatment plants, as well as from the agricultural activities and animal or livestock husbandry where they are. So therefore, interaction between these bacteria along with the antibiotics, as I said earlier, cause the evolution of these antibiotic-resistant bacteria, which emanates from the environment or entering the environment, and therefore it's so much healthy, it's so much hazard, especially the human health. Those interact with the environment, maybe let's say those using water from the rivers, the animal and all that, as well as those interacting with the wastewater systems, wastewater treatment plant sites. Bacteria have developed a number of ways on how they elicit their resistance mechanism against multiple number of antibiotics. One way is through what we call the reduction of cell membrane permeability, whereby they limit the uptake of these antibiotics. Number two is through what bacteria have hydrolysis, especially the beta-lactamases, which they end up inactivating a number of antibiotics. Number three, bacteria are also capable of modifying a number of drug targets, and hence therefore limiting or inhibit the binding of various antibiotics. More so, I can say bacteria have also gained some mechanism of inducing target mutations as well as suppressing various antibiotics. And so much interesting, we have got what we call efflux pumps. These are bacterial efflux pumps. There are 5 majorly known ones. These are responsible for extrusion of a number of antibiotics from their systems before they can release the accumulant effect. And therefore, with all these, they contribute to the resistance that is much investigated and even confirmed by other researchers. So what's the problem? Existence of the antibiotic-resistant bacteria in the environment has led to a number of the gene mutations, and therefore presenting a heavy burden to healthcare system in terms of the increased hospital bills, in terms of their management and all that. It has also resulted to a number of increased or prolonged hospital stay lengths for the patients and even the caregivers. And on top of that, the worst thing is most of these AMR were not early controlled, as resulted to a number of mortality cases. This has created a scientific research gap on the urge of knowing how much or how does environmental pollution drive or foster the antibiotic resistance in bacteria. Therefore, to justify my study. With the lack of evidence-based data and policy decision-making measures of better interventions of addressing challenges associated with antibiotic resistance, it was the need to assess this resistance from the plasmid view of it that will be able to enable us to know, or in order to know the existence of these antibiotic-resistant bacteria, especially coming from our own aquatic ecosystems, and as well identification of these genes, antibiotic-resistant genes, will be able to emphasize on the need of wanting to know more about the drivers, potential drivers of such resistance emanating from this kind of environment. The objective of the study was to identify genes in the plasmids of pathogenic bacteria, fostering or conferring antibiotic resistance regarding various in various aquatic sites, that is the River Sosiani and the wastewater system of Eldoret City. This is the study area. Wastewater samples were obtained from majorly the River Sosiani, the 3 bridges which are highly adjacent and most of them located in Eldoret City. We know it's a busy city, and being a city, much of them is expected to be generated along into the bridges and all that. Other samples are also taken from the MTRH, that is where we had medical wastes along with other commercial wastes from the neighboring sites. Also, we had other samples that were collected from the wastewater treatment sites of Cape Kenya and Huruma Quarry, just adjacent out of Eldoret town. And lastly, we had other samples that were collected from the Eldoret prison for the assessment and the assays of various processes. Under methodology, as I said, these are just part of my the part of my research work. The other 2 objectives I had already done and even published my work. So I can say from the previous study, bacteria, a number of bacteria species had already been isolated and identified. So the continuum of the microbiology aspect of it is whereby these bacteria species from the culture plates were subcultured into liquid nitrogen broth. From there, the plasmid DNA were extracted from these bacteria and quantified on nanodrop as well as on gel electrophoresis. Then there was preparation of 20 sets of primer sequences and the PCR volume of 50, involving all the composition of the PCR, inclusive of the taq polymerase template DNA, the forward reverse primer, as well as the nucleus-free water. Then finally, we proceeded for the amplification of these genes in the bacterial plasmids, and we used the 20 primers of them. Then for the electrophoresis, just to confirm our PCR products, was there any amplification or not. Then I did some of the sequencing just for a few vials, maybe as a regard of the finance and all that, just to confirm indeed these genes are there and in which species exactly where they are. Okay, this is me working on my microbiology aspect of my work, and the other side is molecular aspect of my work. And the results, these are just the agar plates or agar culture plates indicating the various bacteria species that were used in this study. So, as I said, a number of species from various sites within Eldoret City that are sampled, including the Escherichia E. coli, Enterobacter aerogenes, Stobacter frundi, Klebsiella pneumoniae, Klebsiella extoca, Proteus vulgaris, Proteus mirabilis, Pseudomonas aeruginosa. We had Salmonella enteritis and Yersinia enterocolitica. All these, my study detected 10 gram-negative pathogenic bacteria from all the sites that were sampled. Sorry, I think it's not much more clear. But just this to indicate after extraction of DNA, just I wanted to know the size of the plasmid the approximate size of the plasmid that I was dealing with. So it was approximately 2,000 base pair of it. Dipping down in the mediator, the base of my work, the study detected 20 antibioresistant genes in the plasmids of various bacteria from this aquatic environment. And I can say the quinare gene was the frequent gene that was detected in all the bacteria, all the 10 bacteria that I already mentioned at the base pair of 347 the base of 347. Quinare gene induces resistance of bacteria against quinolone class of antibiotics. Number 2 is what we have ADA1 gene. This one confer resistance of bacteria against aminoglycosidic antibiotics, which were detected into the species that I've already listed here. We have a gene known as MPHA gene. It's also a gene that induces bacterial resistance against macrolide class of antibiotics, which was detected in the bacteria which are down there. The study also investigated the presence of black APC gene that fosters resistant bacteria against carbapenem class of antibiotics, and it was detected in the various species of bacteria listed down there. We had also other genes, including the QEPA, TETG, and the SXT genes that were also found and obtained in this study against or in various species of bacteria. On top of that, there's this slide showing blandim gene that was investigated in E. coli as well as Klebsiella pneumoniae at the base of 439 base pair. And this gene has been widely documented or discussed to be the causative agent or against resistance of beta-lactam drugs as well as carbapenem drugs. On top of other results, I may say other genes that maybe I didn't capture their images and all that, but it's a representation of others. We have the presence of BLASHIV gene, induced resistance against the beta-lactams, the F5 gene for the trimethoprim. Then we have other genes that were detected as shown there. We have the SUL1 to SUL3, that's for the sulfonamide. And lastly, for the TET-A, TET-G, and TET-M, you were also obtained this study showing that the bacteria are much more prone, being resistant to a number of tetracycline antibiotics. I went ahead and did some sequence analysis, although not fully analyzed, but I can say that the sequence analysis also confirmed the further presence of the quinare gene, especially in E. coli, Klebsiella pneumoniae, and the TET-A gene in Strobacter frundi. An overview, I can just say, plasmids, bacterial plasmids within the environment, especially the biofilms. We have a number of them. They are capable of acquiring and carrying these antibioresistant genes, which further spread to other pathogens or other microbes, and it is more health risk, especially to us, because with this one there is more projection of development of multi-antibiotic resistant infections, not only to humans but to other aquatic biota and even to animals. I can say my study was furtherly supported by a number of evidence from the researchers, those who detected the QEPA and quinare gene, the SUL1 gene, the FRA gene, and as well as the MPH gene in various species isolated from the hospital effluents as well as other effluents from the environment. In summary, given that having a wide distribution of such genes exist in our aquatic environments and all that, although it has been that not much more documented as is part of the research ongoing and all that, can say Rivers Oceani and the sampled wastewater in questions are the critical reservoirs of antibiotic-resistant bacteria, which are pathogenic and more so endangering the life of the human health and all that. And most of these bacteria, their plasmids all along with that, have shown resistance in my study against a number of antibiotics which are commonly used, and the conventional one especially, which are in markets and in hospitals, or the one which are administered to a number of patients, including the aminoglycosidic, beta-lactams, carbapenems, trimethoprim, macrolides, quinolones, sulfonamides, as well as tetracyclines. And therefore, these just links to what extent can our environmental pollution play a significant role in terms of increasing, or let's say in terms of evolution of antibiotic resistance from the environment against multiple drugs, and how does it link especially to our genomic global world. So what can I recommend? For those people or individuals using water from Rivers Oceani, is a recommendation for them for having the regular treatment of such water because of that possibility of ARGs within the environment. Then there's also need for the steady monitoring of such wastewater systems along with that one. Then for those, there's need also to check the strength and efficacy of the existing antibiotic standard, especially those ones that were synthesized in early centuries, that indicated low inhibitory effects against, or the one that bacteria had shown resistance towards. I can recommend that the government also need to check on better suitable laws on effective waste disposal management and wastewater treatment management, and this might help in reducing a number of antibiotic-resistant drivers within the environment. The future research, apart from just bacteria, they also need to check antimicrobial resistance of other microbes against other drugs. Apart just using the environmental DNA, or you can say apart just using the environmental samples, you also need to advance on using clinical isolates for the same purpose of generating the wide spectrum of MR. They also need to check the chemical and environmental factors existing in this aquatic ecosystem that are the first drivers of MR in various microbes. Apart from that, they also need to do what we call proteomic studies, especially regarding the number of the type or the various antibiotic-resistant genes that have already been discussed in the study. They also need to involve or partner with the bioinformatic way of it so that they can well understood the proteins and all that. I already mentioned part of this work already been published in Microbiology of Journal of Microbiology Spectrum and all that. Thank you all.
Thank you, Sharon, for your presentation. We welcome questions from the audience.
Yeah. Thank you, Sharon, for the good presentation. Just one question. If you were to do this study maybe in 10 years to come, hopefully we'll all be there in 10 years, what do you expect, or what can you project?
Okay, thank you so much for the question. Yeah, you see, science is all revolving around. So I may expect, yes, I know a number of World Health Organization along with the National Action Plan, especially on MR, they are trying as well as to come up with various measures, or even encouraging young scientists to do much on MR, so that at least we can come up with various possible solutions towards. But given that there is this given that there is existence of gene mutations, especially from these bacteria, pathogens and all that, so maybe I expect a new era or a new other existing, the other one might be also to the extent of it. I can say the other genes might also maybe, let's say, because these genes will have the hereditary ones and the one which are just acquired. Maybe at some extent I may say, as a regard of the gene mutations which is happening every now and then, I expect much of it, maybe other new emerging of genes in the question. Yeah.
We have one more questions.
So just a curiosity question. Were certain resistant genes associated with specific sampling sites, or was their occurrence, was their occurrence widespread across the different sites?
Oh, thank you. I can say just a few of it. Let's say, for example, the Quepa gene that induce resistance of bacteria against the quinolone. It was so much distributed all along all the sites, the Quepa gene and the quinare gene. Yeah. But others, they were just in few selected areas of my site or sampling sites. Yeah.
Thank you very much for that nice presentation. Evolution, adaptation, and that means ability to evade any sort of stress. Is there any possibility that this resistance could have been attributed to maybe hybridization, if bacteria are able to hybridize among themselves, or conjugation, or just acquiring this free eDNA in the environment? Did you look at that? Thank you.
Okay, I didn't look at that, but what I can say, in terms of hybridization of all that, what I know is within the environment, bacteria end up forming what we call the biofilms. And there is a way these biofilms, especially I had already mentioned earlier, these antibiotic-resistant genes, they are not only just maybe acquired from the plasmids, maybe from the V of it, but to have, especially those from the chromosomal DNA of it, so they can be during that time of interaction within the biofilm, during the various time of maybe say the survival mechanism of them. There's a way of these genes, especially if one bacteria may, for example, had some hereditary genes from the parental chromosomal DNA, there's a way of it can be able to exchange and be implemented or impacted on the plasmids. And now it becomes spreading from one another to another population. I don't know if I answered you, but
Thank you, Sharon, for your presentation. Let's appreciate Sharon. So our next presenter is Dr. Salina Rono. I must say that Dr. Salina Rono is also my head of department. So it's a pleasure for me to introduce her to present on genomic characterization of antimicrobial and multi-drug resistance in bacteria isolates from Lake Victoria, Nile tilapia supply chain. So Dr. Salina, you're welcome. You have 15 minutes of presentation and maybe followed by 5 minutes of questions.
Good morning. Thank you. My name is Selina Rono. I am a medical microbiologist with special research interests in infectious diseases and antimicrobial resistance, and especially linking the One Health approach, realizing that we now have an interconnection between the diseases that affect humans, a link between the environment, the animals, and the human beings. And we actually are getting a double threat. And looking at AMR, this study was looking at characterizing antimicrobial and multi-drug resistant bacteria from the Kenya Nile tilapia supply chain. We wanted to look at whether the food that we eat, especially the fish, because currently people are moving away from red meat, they want to take white meat. And so is the white meat safe? And we wanted to look at the source and all the way up to the market, tracking that journey to see what are the points at which microbial contamination comes in, and be able to detect antimicrobial resistance. This study is part of a study that has been done elsewhere through the Kenya, UK, low and middle income country. We have studies that have been done elsewhere in Egypt and India, and the team comprises members from the UK, University of Eldred, wanted to see the Kenyan situation. Part of this work was presented yesterday by Sylvia, but today I want to present the one on AMR. So antibiotic resistance is a significant public health problem. We know it has jeopardized effective treatments. We have infections that are affecting humans, becoming resistant to the common antibiotics that we use. And if this is happening, we have seen a record of mortalities of persons who would actually have been treated, dying because the drugs are not working. And so this is a serious problem recognized by WHO. And there is now a new dimension that even the food and the environment, yesterday somebody presented about the environment, and we are also seeing aspects of antimicrobial resistance coming in the environment. Common problems like bacteremia, blood infections, gastrointestinal infections, urinary tract, could be ailments that are treatable, but they are becoming difficult to treat. So AMR is actually a social problem. It's also an economic issue, as shared by Sharon, that the patients, infected patients, spend a long period in long hospital stays, which becomes very costly. And quite a number of deaths that have been realized. So Kenya, of course, through the WHO, which recommends that other countries take action to prevent this. We have the latest WHO report that is an action plan for AMR was in 2025. So it's a very late one that is now fronting the One Health approach, which combines WHO, Food and Agriculture Organization. There is also the World Health, Animal Health, Organization on Animal Health, and UNEP. So it is a quadripartite kind of arrangement, bringing people together, experts, to try and address this problem, because it is not purely confined to humans. Even the pets around us could pose a problem. The livestock we keep could pose a problem to us. So there is a way we have to take measures to really create barriers to prevent this problem escalating. So there is the major recommendation is antimicrobial stewardship, which is a global action plan, which says that every patient should be able to receive the right antibiotic at the right dose for the right duration, and this will be able to reduce both treatment failure and resistance selection pressure. So if that has to be achieved, then there is need to prevent anything extraneous that would come and pose harm to us. So how do antibiotics get into the fish supply chain or into the aquatic ecosystem? Through the feed. In our study, we actually focused on caged fish. Caged fish in Siaya, a place called Anyanga Beach, where there are cages, so many cages. Fish is actually farmed commercially. There is a picture there showing the feed. We actually interacted with the farmers to look at try to find out what they feed their animals with, their fish with, the treatments they give, and all that. So the intensification of aquaculture has led to the increase in disease outbreaks. This has really caused high dependence on antibiotics, even in the fish farm. Of course, in Kenya here, aquaculture was not as developed in the past. I think it peaked around the late 80s, early 90s, when the government injected in some funds through the economic stimulus package to promote aquaculture as an alternative source to expand the economy. So through animal feed, through pollutants that are discharged into the aquatic environment, through industrial waste, domestic waste, all these could release antibiotic residues that are taken up by the fish. So how does antimicrobial resistance come about? We have various mechanisms, like the efflux pumps. The organisms could have efflux pumps that expel antibiotics. And then in other sites, they evolve mechanisms to block penetration of the drugs. Others have enzymes like the beta-lactamases that degrade the antibiotics, and even alteration of the sites. So these are escape mechanisms by pathogens or disease-causing agents to be able to protect themselves against the action of the drugs. So what is the problem? we realized that there is limited data to link antimicrobial resistance along the food supply chain. In this case, we were focusing on fish. So we needed to track that journey from the source to the market, even up to the plate. How is the safety? And is there any evidence of antimicrobial resistance? There's also weak diagnostic capacity, widespread empirical antibiotic use, weak diagnostic capacity. Most of the labs, even in our Kenyan situation, the labs are not that fully equipped. They don't have the right infrastructure to do surveillance studies and try to be able to create barriers to this before the problem of AMR escalates. So we thought there was need for high throughput technologies to be able to screen and to do surveillance and be able to ascertain the status. So we wanted to determine antimicrobial resistance along the Nile tilapia supply chain. And we had a research question trying to find out the resistance profiles of the isolates along that whole continuum. We also had a hypothesis to be able to establish whether there was a significant relationship between antimicrobial resistance at various sites along the chain. So the study was done in cage farming areas of Siaya. There was the harvesting at the beach, the right in the lake, the fish farms in the lake, and then the landing site. And then from the landing site, the fish, it was usually, we observed that it is loaded into trucks for sale in the distant markets. There are these motorbikes also who come to transport the fish to various markets. There are other vendors who come with their containers to buy the fish. But we were able to track particular trucks carrying fish to markets all the way from Siaya through Kisumu to Eldoret. And so we, in the residential areas, from the harvesting sites, it went all the way to the local traders, first to the wholesale market in town, and then we find this many people who come along to buy fish, to sell in their stores, to sell at the market, to sell by the vendors, and even in the eateries and supermarkets. So we had the sampling, sterile swabs, and various sites. The landing site, we collected swabs there. Of course, we collected swabs even at the cages and the cage water and the truck, and then all the way we followed that, and at the wholesale point, the containers that were loaded with the fish, the crates, and all these other materials that the vendors came to collect their fish with. So there was the microbiological processing, and then antimicrobial susceptibility testing. We used the conventional method first to be able to ascertain the antibiotic susceptibility status of the isolates. And then we were able to pick the most resistant and then be able to package them, because the molecular bit, the whole genome sequencing, was done in the UK at Microbes NG. So we packaged the most resistant isolates. And that is, we were able to identify the multi-drug resistant candidates. These were isolates that resisted at least more than 3 antibiotics. So these ones are the ones we picked. Those that resisted 2, we ignored. And we had, we sent about 30 isolates, I think 30 isolates for whole genome sequencing. Of course, the number of isolates were many. And of course, once the isolates were shipped, they were sent to Microbes NG where DNA was extracted using an appropriate extraction kit, then subjected to whole genome sequencing using the Illumina technologies. Then the genomic DNA libraries were prepared, and then they were trimmed using a trimomatic version. The DNA libraries, of course, were uploaded into the gene bank, and then the de novo assembly was done using SPADE's version 3.7, and the contigs were annotated using Prodca. Now, after that, the data was sent to us. The sequences were downloaded from the gene bank and subjected to bioinformatic analysis, and then we were able to get the results and to do the necessary interpretation. Those are some of the pictorial presentations of the sampling sites all the way up to the wholesale market. The results, we have here the heat map of antimicrobial antibiotic resistance by the isolates. And this picture shows the hierarchical clustering that represents the antimicrobial resistance presence across the various isolates. Initially, from phenotypic analysis, we obtained a high abundance of E. coli, Salmonella, Shigella, and Staph aureus. And from this heat map, that is the distribution. The clusters with similar microbial profiles suggest shared contamination sources or environmental conditions, and highlights a unique contamination event that may reinforce further investigation. We have proportional resistance across antibiotics. We had various antibiotics, specifically 13 of them, and majority of the isolates displayed a considerable proportional resistance rating, with a few demonstrating more balanced distribution against across the organisms, showing very low level of sensitivity and intermediate resistance and resistance categories. Particularly, very high resistance was seen for the beta-lactam drugs, oxacillin, penicillin, methicillin. There was high level of resistance, and also for chloramphenicol and minocycline. When looking at distribution of antimicrobial resistance ratings by source, we had the various sources, and you can see there was a level of resistance experienced all the way. It was not just limited to one point, but moving away from the source at the lake to the wholesale and retail points. That is where we found a high level of antimicrobial resistance. The resistance and multi-drug resistant species. We see Shigella at 29.7, Salmonella 17.2, E. coli 25. And then the resistant isolates, high level of resistance was seen in Staphylococcus aureus. And Staph aureus is largely due to the handling, the food handlers, whereas the other three are show evidence of some fecal contamination. Drug resistance and abundance, we saw highest resistance among the fluoroquinolones, followed by the beta-lactams, and then the lowest among the oxalolidosone. Now, this was the interesting bit, the results of whole genome sequencing. Detected 18 antimicrobial resistance genes, showing resistance to various antibiotics. And remember, I mentioned only 30 samples were sequenced. Now, there were various genes conferring resistance to aminoglycosides, to macrolides, to beta-lactams, quite a whole range. And on the other side, we see the range of resistance genes. In this illustration, it shows the top 25 antimicrobial resistance genes per sample. Per sample, meaning individual samples could actually have several antimicrobial resistance genes. And most of the genes were concentrated among the gram-negative isolates, which link them to evidence of fecal contamination. And there was limited on the for the Staph aureus. Now, in conclusion, the study was able to document antimicrobial, antibiotic resistant, and multi-drug resistant strains of bacteria, the Shigella, Salmonella, E. coli, and Staph aureus. And remember, there was a heat map that showed several strains. Through whole genome sequencing, the 4 were detected using conventional methods. But whole genome sequencing revealed actually more than 150 different species of bacteria with varying degrees of antimicrobial resistance. There was a resistance of 23.5% and 75.3% for multi-drug resistance, and 1.2 was sensitive. Yeah, this is alarming really to see that these environmental isolates show high level of multi-drug resistance, indicating that the kind of food we eat, we need to be more careful about it. 29 distinct resistance genes across the antibiotic classes were identified. And most of these were associated with plasmids and integrons, indicating their vast potential for horizontal gene transfer, especially across different environments. And the highest level of resistance occurred at the tail end of the supply chain, the retail markets, the wholesale markets, and the food vendors. We recommend use of high throughput technologies for pathogen detection along the fish food systems. And then AMR surveillance programs to be instituted. Public health education and trainings really need to be mounted, especially for the fish farmers and the wholesalers. And then we also need to strengthen the One Health approach to looking at issues. We have to be careful about the animals that we interact with, even our pets, the livestock, the chicken, the cows, and all this, and really institute safety measures to protect ourselves against infection. Thank you very much.
Thank you.
So we appreciate the Kenya-UK Low Middle Income Partnership, the Academy of Medical Sciences, and the Global Challenges Research Fund, our co-partners in Royal Holloway and University of West London, and the University of Eldoret also for facilitating our project. The cage fish farmers at Tanyanga Beach Shire, they were very cooperative. Thank you so much.
Thank you, Dr. Saline, for interesting talk, well presented and very informative. So we now open the floor for questions, reactions. So maybe I pose the first question. As this seems to be quite interesting, but also alarming. Have you pushed this work to influence maybe policy? Because I think there's something that needs to be done. Have you taken steps such that policy can be done, such that an action can happen?
Yeah, we have initiated the process of just ensuring that this data or these findings be able to get to the policy makers to be able to institute appropriate safety across the population.
Okay, thanks, Saline, for the good presentation. I was just wondering, like, just a comment and concern regarding that this within the food chain, food supply chain. One, did you try to look at the different ways of handling across the supply chain? And if this really form part of the metadata for maybe informed analysis, just to look at if there are different ways of the fish handling after the cage. Then second one, you presented the proportion for species distribution based on the resistance levels. Were you able to get any isolate that was kind of resistant to all the antibiotic panels that you used? The so-called the pan drug resistance.
Yes, thank you. Thank you for your two questions. The first question, you wanted to know whether we did a screening of samples from various sites. So as I mentioned, we were able to collect samples, like at the cage point, and then at the landing site, and then at, where the fish is being loaded, at the loading point, at the truck, and then at the crates, and then at the wholesale market, where these vendors are coming to buy, then followed it all the way to the tables where they are laid, at the retail points. So all that data is available. Yes, we have.
It would just be interesting really to understand the abundance across the food chain, like the supply chain. Just know at what point we have more of star
yes.
present and at what point we have more of this specific species present.
Thank you. I think that's what Sylvia presented yesterday. Yes, she did that. Then about the isolate panel that was resistant, to various antibiotics, that data is also available. Yeah, that's why we have recorded multi-drug resistance among a number of species. Yeah, thank you.
Thank you so much. In one of your conclusions, whereby you stated some of these genes were much linked or associated with the plasmids. So maybe I was just thinking, maybe if you have thought of going ahead, I know it was not one of your objectives in your study, maybe going ahead and from just the same samples or the same sampling, you can be able to maybe assess and investigate presence of maybe antibiotics so that you can have a major rule out of being one of the driving factors maybe in this kind of resistance towards within the fish or in the lake.
Yeah, thank you, Sharon, for that question. Yeah, I think that is something we need to explore, and that's homework for Sylvia as she advances to her PhD. She needs to look into that. Thank you.
Okay, thank you, Doctor, for your presentation. I have two questions that are more or less interlinked. You say that your samples were from cages. However, at the market level, we have fish from fish farms, that is from the pond system and all that. And we understand that when they get to the markets, there's more or less no segregation of the sources. So with the risk of cross-contamination, were you able to put that into consideration, the aspect of cross-contamination of this from fish, from farm system, pond system, and the cage system? And two, do you think that fish from these results could be applied to fish from the from the pond system? Thank you.
Thank you. Thank you so much for that question. The first one, we were interested in the caged fish, and we were able to track the journey all the way up to the retail market. And even the point where the vendors came to buy, we identified the vendors and followed them to their business premise. So kind of the data, of course, we have seen that at the wholesale and retail markets, that's where the highest level of contamination was recorded, and even the resistance is seen. Of course, we cannot rule out the contamination emanating from fish from the local sources and the handling. but we were of course we did not label the fish, but we were able to track it all the way from the vendors at the wholesale market. Your second question, I think I didn't get it clearly, kindly.
The second question was, could these results be, could it also apply to fish from the pond system?
Yes, definitely it can apply, because it is just a clear indication that this problem could exist anywhere, and the need to really take precaution. Thank you.
Thank you. Let's appreciate Dr. Salina. And with that, we come to the end of sub-theme 5. And next is we have a tea break. We are a little bit short of time, so we can take around 20 minutes to take our tea. And then we come back to then session 6. Thank you.
To the last session of this symposium, and this is on the sub-theme Genomics in Diagnosis, Surveillance, and Management of Diseases and Epidemics. We will start off with this session before lunch break with 2 of our keynote speakers. And the first keynote speaker is Professor Anise Api. I believe she's in Nigeria, or she'll inform us where she is. I'll give a brief introduction of Professor Anise before she gives us a keynote presentation. Professor Anise holds a PhD in veterinary clinical pathology from University of Ibadan, Nigeria. She also has held a postdoctoral research fellowship at Harvard University, USA, and several other fellowships. She also has taught previously at the Department of Veterinary Pathology, University of Ibadan, Nigeria. And she currently serves as the Deputy Director of One Health Research Zoonotic Diseases Surveillance at the Institute of Genomics and Global Health at Redeemers University, Nigeria, where she's leading significant research initiatives. So today she'll share some of our research projects and initiatives, and the title of the presentation will be on harnessing evolutionary genomics for comprehensive disease surveillance and pandemic preparedness through a One Health approach. Professor Anise will be presenting virtually. Professor Anise, can you hear us?
Yes, I can. I can hear you. Let me put on my video. Thank you very much for inviting me. I say good morning, everybody. Good morning, ladies and gentlemen. I want to start by apologizing for not being able to attend the symposium, the regional symposium, physically. I wish I could be there in person to interact with the great member of this symposium. But unfortunately, I'm now in Cameroon, where I lost my very close cousin. So that prevented me from being there with you. But I'm with you in spirit, and I have tried to follow some of the presentations. So that being said, I have been introduced, and I want to thank the organizers of this symposium for allowing me to share some of the research work we do using evolutionary genomics to address public health diseases. On that note, I want to start briefly by what led me into this sphere. I haven't taught in veterinary pathology and then practicing my veterinary profession. So in early 2018, Nigeria experienced one of its largest Lassa fever outbreak, and cases surged in multiple states, and a healthcare worker became infected and critically ill. Let me share my slide. I think I'm not sharing my slide. I can't see my slide. I can share, sorry. And I want to put it in the slide mode, okay? Can you, can you see my slide? Okay. There is a on slideshow. I don't know. Okay. Can you see now? Okay, with my story, I said that in 2018, Nigeria experienced one of the largest Lassa fever outbreak. And cases such are called multiple states, particularly Ondo, Ebonyi, and Edo states in southwestern and southeastern Nigeria. Well, healthcare workers became infected, and then there was a critical question emerged as the disease spread rapidly. Then the question was Lassa virus now spreading efficiently from human to human? Because if that was the case, the implications were enormous. Because if there were sustained human-to-human transmission, that could signal the beginning of a far larger epidemic and regional and global consequences. Yet the national epidemiology alone could not address this question. So then the genomic changed the narrative because we needed to understand how the disease was spreading. In so doing, by sequencing viral genome across affected regions, scientists reconstructed the evolutionary relationship of the virus and revealed that most infections were not driven by human-to-human transmission, but by multiple independent spillover from the reservoir, which are known to be rodent. I will call them small mammals, occurring simultaneously in a multiple. Then genomic now help to separate fear from evidence. These findings shape outbreak response strategy, strengthening surveillance of also rodent control rather than focusing on human-to-human containment. Therefore, the 2018 Lassa fever outbreak became a powerful demonstration of the One Health concept, pathogen emergence driven by the and the dynamic of the pathogen allow us not only to detect pathogen, but to understand how they evolve, spread, adapt, and potentially ignite pandemic in an increasingly interconnected world. Can you hear me? Yes. I just wanted to share. I don't think you heard me. I just wanted to share with you what led me into the evolutionary genomics for preventive and control measures. Yes, first slice. Yes. Let me try and can you let me stop again and share it. Can you see my slide? Let me reshare it. Hmm. Can you see it now? Is he on slide? Is not on slideshow? Wow. I think the internet is very slow. Back to the first slide. Can you see my slide? Hmm. I'm sorry, my internet is very slow.
Evolutionary Genomics and Precision Medicine in Africa, Opportunities, Challenges, and Ethical Frontiers. Let's welcome Dr. Chororei for this keynote address. Thank you.
Thank you very much, Dr. Tuwei, for that kind introduction, and thank you all members. I really want to thank the committee for giving me this opportunity to give a little bit of my experience from in genomics. I'm really happy because when we talk of University of Eldoret, that is where I did my undergraduate. By then it was Moi University. So I don't know if I should say I have a Moi University's undergraduate certificate of University of Eldoret. But Chepkoilel is home to me. That is where I did my undergraduate. And then later on proceeded to School of Medicine, Moi University for MSc in immunology. And then later on to Maseno, as has been said. So today I'm going to give you a talk on evolutionary genomics. So, when I talk of evolutionary genomics, it's not that I'm a geneticist or anything, I'm an immunologist. And I was really looking forward for Happy's presentation because she would have really covered a lot of evolution. So what I'll give is just a little bit of what I know about evolution, but and then we go to the other parts. So, I will expect us to look at the role of evolutionary genomics in modern medicine, appreciate Africa's unique genomic diversity and its scientific value. I know we have a lot of geneticists here, so you will help me when questions come around diversity of human race. And then opportunities for precision medicine in Africa, that is close to my heart. And then reflection on ethics, legal and social implication. It's also something that I'm really passionate in. And probably inspire the young generation, the researchers, innovators, and leadership in genomics. So to start with, I want to start with this quote. Africa is not only the cradle of humankind, it is also the cradle of genetic diversity. And from what we've looked at, I'm really impressed that not only in human, but also in other species, like fish from Turkanas, seems quite diverse compared to others. So the future of precision medicine cannot be complete without African science, African researchers, and African communities, because this is where diversity is. Now, this is an AI-generated evolution of humankind. I'm not an expert in this, but looking at the oldest, sorry for the small writings, the oldest is fossils that were found recently in Chad. We used to say the oldest were found in Kenya and then South Africa, and now it is Chad, where they have this species or human race called Sahelanthropus. Jadensis, which is 7 million to 600 million years ago. And that was the time before even the tools. And I think this particular species is grouped as human because it was starting to stand upright. And then over time, we have Oronin tugenensis, which was actually in Ethiopia and Kenya. And then it has continued up to the current the current race. Yeah, and I like it because it's not showing, it is actually showing African, not using the whites. Yeah, you can see that from the fourth, that is Australopithecus. That is when they began the tools, using the tools, and then Homo habilis also used the tools. And by the time we get to Homo erectus, they are kind of also spreading to the other parts, Europe and Asia. And then the current species, the Homo sapien, uses tools, smaller brain, and also there is a global networking. Now this is what the AI kind of not actually predict, but it is a trajectory to the future. That in the future we'll have enhanced humans. And then this enhanced human will be, you see what is happening with their brain? Oh. Yeah, around here, there's a lot of chips implanted, and then genetically adapted humans will come maybe in 200 years from now. And then you can have what you call cyborg humans, and then interplanetary humans. And by the time we reach this time, that is how many years? 20,000 years to come, we'll not be talking about women, ladies, the sexes. We'll have the transgender humans and maybe post-biology beings. So this is what technology can do. This is just an hypothesis, but it's possible that such things can happen. And then in the future, humankind, I don't know what it will look like. If we all believe in the story of evolution, such a thing can happen. I don't want to go into the details. Evolutionary is not as linear as it appears. It has so many diverging, and you can see that most of our forefathers are actually extinct. All these are extinct. The only living one is the Homo sapien. And then with technology, of course, we have the biological genetics pathway. There are so many pathways that are possible. We have the biological genetics pathway. And then with the biological genetics pathway, we see what is happening with CRISPR now. People are really editing their genes. So we'll have people that are kind of introducing self-evolution. And then when you go to the next step, we have robotics, a lot of robotics, which is starting to begin. And by the time we reach post-biological pathways, I don't know what will be happening. But all in all, this diagram synthesizes evidence from paleoanthropology, evolutionary biology, and future studies to illustrate human evolution as a branching, not linear process, shaped by mosaic trade evolution and environmental pressures. One of it that we can appreciate, all of us, is the global warming. It's doing a lot in our genomes, with speculative future trajectories informed by emerging biotechnology and cybernetics. So I think I want to mention a little bit about the African genome diversity. And I was happy in the morning there's somebody that was presenting something on how to understand the human genome, because that is the way to go. And in Africa alone, we are very diverse. You see these colors also suggest that we have diversity in that manner. So in the northern, in Africa, we have over 2,000 ethnic groups. And when you say ethnic groups, those are the people that have stayed together, lived together, migrated together. But if they talk different languages, it means that they've stayed apart, and therefore they could be having some uniqueness in them. So over 2,000 ethnic groups and thousands of languages means a lot. And in the north, you can see that there's a lot of diversity in West Africa. Yeah, in East Africa and even to the South Africa, there's a lot of diversity. So there is more variation if you look at even not only genetics, but even the culture that is in Africa. And therefore, sometimes when I was doing some analysis for breast cancer from Kenya, we cannot use Yoruba reference genome and say that this is African, because East Africans are very different from West Africans. And there's some Bandus in South Africa that could be similar to some of the West African, but not entirely the same. So in Africa alone, we are very diverse. And this diagram also shows how intensive that diversity is. Each and every color means a diversity. And in the north, you can see that the height of diversity, it is lower in the south and in the north. But in the Western Africa and Central Africa, it's high. The genetic diversity is very high. And these are migration patterns. This is what I would really like to look at. I don't know if people are reading this. It's not so clear. Is there a way we can improve that? The focus? Sorry, most of my slides will be like this, really busy slides. But it's okay, I think we can get a better slide. Okay, I think that is the best you can give. So, most likely this is not what I wanted to share, but I think it's still okay. So what inspired me to genomics is work from a Nigerian oncologist called Olopade. I don't know if any one of you have read about Olopade. We now have immune-based therapy called by a name. So Olopade is an oncologist and also a geneticist who worked with the University of Chicago. And she felt that African genome or African population could be having a diverse or a unique genetic makeup. The reason why she came up with that hypothesis is because she saw patients from African ancestry, especially the cancer patients, having aggressive forms of cancer, especially breast cancer that she started with. So patients with African ancestry seem to have aggressive forms of breast cancer, and that is true. Even in our setup, we have very, very lethal breast cancer. People are dying of breast cancer in Africa, although the prevalence is not that high compared to the Western world. Breast cancer kills in Africa more than the other world. There are other things like, for example, maybe our infrastructure is not better. There is delay in diagnosis, but that alone will not explain the very high mortality. The mortality rate, I think, is over 75%, compared to less than 25% in the world. I worked with South Africa, and I also witnessed that people around South Africa, especially in Cape Town, live long with cancer, but we die of cancer. Take, for example, a cancer called triple negative, breast cancer called triple negative. It doesn't have ER, it doesn't have PR, that is estrogen receptor, progesterone receptor, and HER2. It doesn't express the receptors that are targeted. And therefore, if you have triple negative breast cancer, especially in 10 years, 5 years ago, you would easily die. And it is a form that is seen in younger generation, slightly younger people, and also it kills. So Olopade thought that maybe it is because of the genetic makeup that we have as Africans that is making us die of cancers and any other diseases that are aggressive in our population. And therefore, that really motivated me to start thinking about genomics. So when we look at the implication of African, going back to the implication of African genetic diversity, African genetic diversity has play a very important role in pharmacogenomics. This is when you understand which drugs works for you based on your genes. So I'll mention a little bit what we've done about that, and then enhances our understanding of gene interactions, gene interaction together with the environment, helps in mapping of disease genes through comparative genomic studies. It enables validation of genetic biomarkers that if we participate in studies that are discovering biomarkers. But if African is underrepresented, then we may not be capturing the biomarkers that are of use to the African race. So it can also help in creation of updated genomics reference panels, which at the moment we don't have. I think the group that works with the presenter in the morning are now developing it, and then also develop discoveries of novel genetic variants. So we expect a lot of these novel genetic variants because of the diversity. So this is informed by this study, a study done by Sibomana, and they did a very comprehensive review from PubMed, Google Scholar, African Journal of Journals, and so on, and they use these keywords. Okay, so I want us to think of this story, the African ancestry. Is it an illusion? So imagine two patients, Aji from Nigeria and Kofi from Kenya, both being treated with the HIV with the same drug. And to a doctor that sees a one-size-fits-all approach, he would see these two patients as simply Africans, and therefore they should be treated the same. However, Aji thrives on medication while Kofi develops a life-threatening hypersensitivity reaction. What has happened here? Why is it? The reason is because the genetic makeup that predisposes to this hypersensitivity reaction, that is HLA-B 5702, is virtually present is virtually absent in Nigerians, but you find it in 14% of Kenyans. So Kenyans are likely to react to this drug, just because Kenyans are very different genetically from the Nigerians. So this story proves that Africa is not a single genetic category. It is the most diverse population on Earth, and ignoring this diversity can be dangerous. So why evolution genomics? Evolutionary genomic studies have genomes change over time and how human populations adapt to their environments. You can imagine the environment that was experienced like even 100 years ago is very different from what we're experiencing now. There was no global warming as now. So the key concepts include human evolution, genetic variation, natural selection. I've heard people talking about this in fish. Adaptation, population genetics, and disease susceptibility. There is also the African genome contains the greatest human diversity globally, as we've said, ancient and complex population histories. And these histories would influence a lot of genetic makeup because your environment, what you eat, your lifestyle can also determine, can influence your genetic makeup. Important clues of understanding disease and treatment response will come from Africa. So examples of relevance to Africa, we have sickle cell trade and malaria. Examples of the natural selection. So over time, it's like people that were having the sickle cell trade and have the ability to resist to malaria, the ones that seems to be surviving. So you can ask yourself, what will happen if this will continue for a very long time? And then we have things like lactase persistence, adaptation linked to pastoralist communities, which they have the ability to digest milk in adulthood. And then even with the infectious disease resistance, I think this one has been captured so well, where you have genomic variation effects, susceptibility, and immune response. So what I'm seeing, you people, you've done a lot of antimicrobial resistance, but I think there is importance of translating it into human or clinical impact. And then transitioning statement, evolution shaped our biology, precision medicine aims to use the knowledge to improve healthcare. So this is just demonstrating how the evolutionary adaptation and health examples relevant to Africa. We have the sickle cell trade, protecting against severe malaria. This is due to natural selection in malaria endemic regions, increasing the frequency of sickle cell trade. And then we have the pastoralist with the lactase persistence, adaptation to ability to digest milk in adulthood, of which most of us may not be having that gene already. It is a genetic adaptation that provides a nutritional advantage in milk consumption or consuming communities. And then we have, of course, infectious diseases. There's a lot that has been done in HIV and malaria, which genes are responsible to predisposing risks and which ones are protective. So evolution has also equipped a population with different genetic tools to survive diverse infectious challenges, including the ones that we are seeing in this room today. So our immune system will try to be able to adapt to the resistant variants that we see in bacteria and many other microbial agents. So evolution shaped our biology, but precision medicine has come so that we use what we know to improve healthcare. So this is what I was talking about. My work was inspired by Olufunmilayo Olopade, who this lady or doctor demonstrated a remarkable genetic diversity of African population, and he linked this to aggressive biology of certain breast cancers. This realization inspired my passion of genomics, strengthening my commitment to advancing African-centered genomic research, precision medicine, and equitable care solutions. And these are the areas. Some of these questions have not been answered. This was in 2013 when I was writing my PhD. And that is what I did, characterize breast cancer, and later on did genomics. So this is what I did for my, not PhD, but postdoc. So after my PhD, after characterizing using immunohistochemistry, I proceeded with the samples that were collected from 107 breast cancer patients. I took them to Stellenbosch University, where I did extracted DNA from the saliva samples, and it has a very interesting story, because I remember when I was collecting these samples, I had no fridge, I had no money. So what happened is that I read about a company called Genotech. They were making saliva collection kits, and I wrote to them, and they gave me advice. I was even asking them, do you know of anyone who is using these saliva collection kits? And they mentioned to me that there is only one person in Africa that is using that currently. And that was Maritha Kotze at the Stellenbosch University, Tygerberg campus. So that is how and then I asked them, can you connect me to them? That is, I was very young, and that is how I got connected to a person that actually funded my postdoc and also helped develop my passion in genomics. So we did that. And we were looking at the family history because we wanted to do genetic testing, and we realized that out of the 107, some of them actually declined giving the consent. One of it was a very young girl, very young. I think she was 18 years. And she told me that she was raised by a single mother, and she had nothing So she had to sell herself to get fees, to get to school, to get what they eat. And she developed breast cancer, but she feared getting into the study. And actually, she ran away from the hospital, MTRH. And then the others are some of the men that we approach, but men are not really free to talk about their privacy, and when they have breast cancer, they don't want to talk. So there are a lot of challenges also in doing research in genomics, because it really gets into people's private lives and knowing more about them. So this is what we got, family history of 15. So it is not ethical to do genetic testing in anyone. You have to have a reason. There is NCCN guidelines. So the first reason was to do genetic testing for the 15, and then we didn't have enough DNA. Remembering this is saliva that stayed with me in room temperature in my office, just in a box. There's a success story that I wrote with the company. If you look at my name, you can find it. So it stayed for several years, but it is really amazing that we could get enough DNA to do all exome sequencing after some time, except for these two. And then from the ones that we did, we had one patient that actually was lumino B, that is having ER/PR positive breast cancer. And she had a BRCA2 mutation. This is a pathogenic mutation. When you classify using ClinVar and other tools that you use in bioinformatics to classify variants. So this one, this is what we did. This is whole exome sequencing, and this is the reference genome. I think we used the 38, HG38, and this is what we got. There was a change from A to C at this position, causing a stop codon, meaning that BRCA gene was not expressed in this patient, and therefore DNA damage could be allowed to continue. And before you do anything, you have to do Sanger sequencing to ascertain that this is not an artifact. So we did Sanger sequencing, and this is what we got at that position. You can see this is the standard, and for the patient, you can see that the curve is smaller, and there are two, but the normal case, it should be one, a smooth one that is of equal height with the others. So then the challenge came. When I was enrolling patients in the informed consent, because ethics will always ask us to have an informed consent. In the informed consent, it was written that we'll come back to you if we get any of the results that are of clinical importance. And now the next challenge was, we cannot hide this. We have to feed back this information to the patient. But we didn't have any roadmap. So it was a challenge, but eventually we came up. First of all, before we go to that, this is me and the Central Analytical Facility of Stellenbosch. We used ion torrent, and this is a pipeline from the saliva sample. We did the sequencing, FASTQ for quality reports, and then clean the FASTQ, and then do the reference alignment to get the SAM, BAM files, and then the variant calling. We use several things actually here. We got the VCFs, and then now we subjected to the tools that were giving us the relevance of the variants that we are getting. So this is what we did. We had to give back results. So what happened is that we had genetic counselors. At that time, we didn't have any genetic counselor in Kenya. So what we did, this is before Corona, we did Skype with a genetic counselor from South Africa and a treating clinician here at MTRH, Chandaria. So we had a session with a patient who came with a son. The son was really anxious. He really wanted to know, if this is my mother carrying a mutation, what does it mean to me? Will I get cancer? Should I get tested at this time? That time he was only a teenager, and we advised him that it was not the time to test. It was still early for him to do the testing. So this is what we did for the testing. First of all, review the informed consent. And actually, it shows that the patient signed the informed consent. That is very important. human genomics has a lot of ethical issues. You cannot separate genomics with ethics. So the other thing is that she was ER positive, PR positive, but HER2 negative. And the breast cancer was diagnosed. I think it was invasive ductal carcinoma. And then the next step was to confirmation of the WES results. So when you get the sequencing results, sometimes it could be an artifact. So to ascertain that, you have to do Sanger sequencing. And then the next step was interpretation of genomic results in the context of clinical information available in the research database, and that one we did, and got this variant. And then follow-up studies and clinical management of familial risks. So this is still ongoing because when we canceled the patient, she has several sisters. That time she was 51. That was in 2021, if I'm not wrong, or 2019. She had siblings, sisters, and brothers. And you know, this is an inherited variant. So anyone that has it has a 50% or even more with age of developing either breast cancer, prostate cancer, and also ovarian, pancreatic, that syndrome. So, so she has family members, but not even the daughters. She has also daughters who have children, but only this son came with her. The other members were not interested, but we told them in case they need any counseling, they can come. So, and then we had to document the feedback. The treating clinician by then said that we need to keep that report on the patient file for any clinician that is treating the patient to tell or to use. So I'm really happy that this patient was diagnosed at, I think the breast cancer was stage 2. She's still alive. She has finished her chemotherapy. We don't know if she's cancer-free, but she has been leading a very healthy, normal life. So this is what a paper that we are developing on this experience of having a genetic counselor from South Africa. This is the mother and the son. So this online genetic counseling, there was a referral process, as I've explained. And then there is a very important tool that my mentor in South Africa has done through a company called Genomics. She has developed a very easy way of bringing together these complex data and adding with lifestyle, environment, and pathology. She came up with something that I'll spend some time to explain called pathology-supported genetic testing. Because genes alone will not tell you much. You have to look at it with the biology, that is the pathology of the cancer, for example, and also the environment, where the patient stays, if the patient is a smoker, if the patient is obese, or if the patient is getting eating well or not. So that was followed by the counseling session, and this was before Corona. Now everything is in Zoom. So we did Skype. That time there was no Zoom, and we were able to do that. And then the patient's response were also taken up. And so that is what I did. Now, because I did only 12, yet 13 patients, yet I have data for 40. We did more sequences in another 13 and then 37. Another 37, 27 to make it 40. So we did another whole exome sequencing based on the next category. The first one was family history, and then the next category was if the breast cancer was seen at a younger age, or if there were other things that could justify another genetic test. So we did another 40. And when I came back, I could not do much with the data because I was not a bioinformatician. Then I talked to people from ISIPE, Caleb Kibet. Caleb Kibet recommended an MSc student from Uganda, Makerere, who had benefited from, they have a name that they use. This student was sponsored, and we supervised together with Gerald Mboya from Uganda. Somebody is from Uganda. So this is what he did: non-BRCA breast cancer, predisposition, variation, and identification. And if you can look at his pipeline, it's similar to what we did. But we are really getting very interesting results because we are getting a lot of voices. One, this is a variant of uncertain significance. This is because the reference genome that we are using may not be actually tallying. So according to the softwares and tools that have been developed, because African population is so small, I think we are at what percent now with the whole genome? I think less than 17%, or even less than 10%, yeah. So our genome is underrepresented. So whenever you lay down your reference genome against the patient, you always see unique things that the tools will be confused. And they are confused because they don't know how to classify it. So they classify it as variants of uncertain significance. Otherwise, it would give you as a benign, which means there's nothing wrong with it, or likely benign, and then likely pathogenic, some percent of causing, or pathogenic, and also VUSes, variants of uncertain significance. So with our pipelines, we get a lot of this from African population, which can be reclassified as more data is accumulated or is more, when more data becomes available. So this variant can move to either pathogenic or likely benign. So what we came up with is that we have a lot of pathogenic mutations. And actually, I'm now preparing to go back to South Africa and do, because that's where the DNA is, to do Sanger sequencing for each and every one of these to see if they're actually pathogenic or not. And we are also getting a lot of VUSes. And what is interesting, 9 of these VUSes are really unique. We could say these are novel because it's not anywhere, but they, by all aspects, it's actually looking like a true variant. So a multi-platform genomic approach, integrating West bioinformatics analysis, whole genome sequencing, long-read sequencing assays, and Sanger sequencing validation is currently being employed. So this is what we'll be doing. We'll also try to do long-read using nanopore, and just to see how we can classify these variants. So this is another student, but now this student is, he graduated last year, he's from Tanzania. He looked at my work and he really wanted to do the same, but now with prostate cancer. So this is what he got. He looked at several genes, and I think they sent these samples to Inkaba. And this is what they got. You can see the VUSes are many, and then a few likely pathogenic and variants. But this was targeted. Again, a lot is left out. So moving to what has really inspired my genomics in Africa and really playing a role in advocating for genomics in Africa is what my mentor developed, the pathology-supported genetic testing for application in breast cancer, pharmacodynamics, family counseling, lifestyle adjustment, and change of medication. Because over time, we can be doing this science and talking in boardrooms like this, but if this does not translate to a common mwananchi, then we are wasting resources and also losing lives. So in this way, she came up with a very simple way, not very simple, but simplified way for scientists. So she sits in the middle here with her company, and you can do several things. So if the patient comes, then the patient is referred. This is a patient with breast cancer. And then there's an online questionnaire, or you can give it face-to-face, and then get into the system. And then that system will also direct you on which medical tests should be done. A tumor will have to be taken in, and also the history. And if possible, germline mutation, blood or saliva is taken in. And then after that, after the testing, the results go back to pathology-supported genetic testing, because she has developed an engine that puts together all this information to release a report And that report will go back to the patient or to the genetic counselor or to the treating doctor. And the genetic counselor will now counsel the patient depending on the results. If there are mutations, if there are variants of uncertain significance, it's important to tell the patient. But before you tell the patient, you ask the patient if she really wants to know or he really wants to know or not. And then after that, you can go to 7. That is the new insights and concerns are addressed. This is when you decide if you need to test the other family members because they could be carrying the same mutation or they need counseling and follow-up. And that one will also continue to number 8 and number 9. That is, if the patient comes back through counseling, then some automated reports will be given back to the system. And then if, for example, something is not informative, then you can recommend for all genome sequencing. Like there are so many examples. There is a family that kept on, there are very many members in the family that have a cancer, but none is actually recorded as, is in the system, in the pipeline, or one of the classified variants. So what happened is that the members from 3 generations from this family were sampled, and then they looked at their DNA, and they realized that there is one thing they were sharing, and now that one is going to be classified as one of the variants. Sometimes you may not have what is already discovered. So that family benefited from people putting together all the information from different families and also tests that have been done over time to enable intervention like doing more assays. So again, this is a paper that we are working on. Unfortunately, it is not seen. We want to see if this can be used in the entire Africa. So we are working with somebody in Egypt. I am in Kenya, and Maritha and other people in South Africa. So we want to face the challenges of feedback of testing results. How should we feedback, especially results that come in research? Sometimes these tests are very expensive, and you cannot only use as research and stop at that. There must be something that should be done. In case you find a variant of clinical significance, will you leave it at that point? It's not ethical to do so. So then she's coming up, we are coming up with this pathway to try and address that. So going to precision medicine. So what is precision medicine? You can define it in different ways. Moving from a one-size-fits-all model to account for population heterogeneity. And there are many studies that are coming up now. The G1 studies with polygenic risk scores are going to be really informative. Tailored interventions, which customizes prevention, diagnosis, treatment based on individuals' unique profile. And the core goal for precision medicine is to deliver the right treatment to the right person at the right time. Yeah, that's a very busy slide, but it all brings together the 4 pillars of precision medicine, which is environment, lifestyle, social determinants, the culture, and so on, and also the genetics. So genetics alone will not play a role. You have to consider the environment. There is a lot that is going on with microbiome. Then the lifestyle is the person eating well, leading a healthy, active lifestyle or sedentary lifestyle, and then the culture. So all these are factored in. So this is a summer school for a personalized medicine conference. And during this summer school, these are people that are funding the project from the EU. So EU and Africa are coming together for the personalized medicine standard development of standards. And this was in 2023. I attended this conference. This is Maritha, and then these are people from the other parts of Europe. So personalized medicine, according to this body, refers to the medical models using characterization of individuals, phenotype, genotypes, for tailoring right therapeutic strategy and for the right person at the right time, and or determine the predisposition to disease or to deliver timely and targeted prevention. So this is how this body defines it. And there are very long ways of which they suggest for training in Africa in personalized medicine, encouraging a lot of clinical trials, healthcare policy and economy. So a lot is going on here, and basic genetics and genomics, and also things like bioinformatics, which is really growing in Kenya and other parts of Africa through H3 Africa and also other bodies that are coming up. The issue of biobanking is very important. This is what we should embrace in genomics, and communication and patient engagement. So understanding the communities, the culture, and so on is really important, because in Africa, informed consent is sometimes communal. People will say, I want to know the results if my husband agrees. I can't do mastectomy unless I go back to my family and ask. So there's a lot of community engagement that is required. So harmonizing precision medicine, you have your genomics, clinical implication, diversity, ethics, and culture. Sorry. So in what applies most in Africa, currently there's a lot of cancer genomics. There's a lot with infectious diseases, HIV, tuberculosis, malaria, and now antimicrobial resistance, rare diseases are also being looked at, immunodeficiencies, but of importance is understanding the pharmacogenomics, which drugs works for you. The patient that we were giving consent, we also did some pharmacogenomics on her, and we realized that she has a gene that is called CYP2D6 allele 17. So this gene will make you, if you have that allele 17, it means that you are a slow metabolizer to tamoxifen. And she was on tamoxifen, actually. But luckily, she was transitioning from tamoxifen to aromatase inhibitor because she was crossing 51 to 52. So breast cancer patients are given aromatase inhibitors if they are post-menopausal. So, but if we didn't know that, you can continue African population genomics projects. I think one of them is the one that was presented. African population genomics projects. I think one of them is the one that was presented in the evening. National biobanks, African-led bioinformatics training programs, and I'm sure some of the students here are also interested in that. There is one aspect that is still lacking, genetic counseling. It's very important, and also using the non-animal models. Apart from using rodents, you can use the on-chip organ and organoids and the technologies that are really bringing a lot of change. So this is what H3 Africa has done with us, Moi University. When I came back and I was not able to analyze data, I went back to one of the PIs of bioinformatics in H3 Africa, that is Nicola Mulder, and I told her that please help me train people in Kenya. She allowed me to be a host. Moi University hosted this African genomic medicine training, I think twice for 2 years. This we have been training. I think some of you benefited from it, or in this class, either this or the other one. Introduction to bioinformatics, next generation sequencing, which was done once, and also genome sequencing bioinformatics. So we've been working together, and we are happy to even expand to maybe having MSc in bioinformatics and other things. So my current focus is to still look at improving breast cancer outcome among the Kenyan women through precision oncology and personalized therapy. So the problem is that even though we have chemotherapies and we have many other therapies, we still develop resistance to those chemotherapies and toxicities. Some of the people die not because of the treatment, but because of the toxicity of the treatment. It's because your genes will not metabolize it. Instead, you die of it. So we have a new study in collaboration with Linköping University where patients sample, this is fresh sample. So we sit at the theater when the breast is removed from the patient. We take in 15 minutes to pathology lab, take a piece from the tumor and put it in certain RNA lather, and then that will be used in these models. So the first, this is a non-animal model. This is the multi-omics. So we'll do organ on chip, and then zebrafish model xenograph. So this is where we have a fish. I have a fish also in this. And I'm happy that there are very many people that are doing things with fish, and I'm now confident to even ask these people to give us this technology, because we have experts that can actually rear this special fish for us. So what we'll do, we'll put that organ into this fish, zebrafish larvae, and in 5 days you can tell 2 things, or several things. One, you can tell if the cancer will metastasize or not. Then we come back to the clinician and tell the clinician, please, this is an aggressive form of breast cancer. You need to treat it aggressively. We'll also test several chemotherapies that are used in MTRH, for example, for these patients. And then we come back in 5 days and tell the clinician, this chemotherapy will not work, this one is likely to work. So we are doing a pilot study on that, and we are hoping that this will change the patient outcome, because that is our target. We want people to live longer and even prevent this if possible. So this is the zebrafish. Why zebrafish? Extrauterine development, fast embryo development, and these are special zebrafish that have been actually humanized. You can do so many things. It's transparent. You can see. This is where you plant the tissue here, and in 5 days, this tissue is given a color. In 5 days, you'll see even the color gone. Or if the tumor will not metastasize, it will grow big. So it's a very special fish, but it's reared in the aquarium. So it can tell us about the early efficacy data, metastasis insights, combination therapy. This is another thing that is ongoing now. If one drug is not working, can you combine 2 and see if it is working? So this is what doctors are trying, and then especially with resistance, and then responders versus non-responder patterns, translation biology evidence, and mechanism validation. So I'm happy that our samples will get into the Europe when they are discovering biomarkers and also testing or validating these biomarkers. So challenges faced in Genomics Africa, I think this one has been mentioned. The first thing is infrastructure challenges. I don't think we have sequencers in Eldoret that are working, not in the, unless you have it in University of Eldoret. I've been talking about getting a sequencer since I came from South Africa, but there are many sequencers in Nairobi now. So the other thing I really wanted to tell the speaker in the morning is there a way they can do devolution for genomics so that they bring some of the things to Eldoret, because this is what we lack. We have patients, those who've been to Chandari have seen. Cancer is really a reality here, and we could do much if we had infrastructure. And then human capacity, shortage of trained genomic scientists, limited mentorship opportunities, and then there is a problem of brain drain. Our medical doctors, they want to get out as soon as possible. And these are the top cream of our country. So if this continues, what will happen? The government of Kenya has used money to train them for 6 years. They are very good doctors, but they are going out to be used by somebody else. So this is a real problem in Africa. Data challenges, underrepresentation in genomic database, we are very, very low. Data sharing barriers and limited interoperability. And then the clinical translation challenges, integrating genomics into healthcare system. Yeah, we have to really integrate it to healthcare system. And then limited awareness among clinicians. So trainings are ongoing, even among our clinicians. Okay, and then for ethics, I don't want to emphasize much. What ethics and genomics, you cannot separate them. There is a need for justice, sovereignty, and trust whenever genomics or data of genomics is concerned. And then engagement for future vision. Translation into local languages is important. That is, the informed consent can also be translated into the languages. most of the time when you're talking to people, they just say yes because they're either too sick or not too sick, but they are sick. They just want to say yes. They sometimes feel that it is part of the hospital routine, part of the services. So they decide just say yes. And then sometimes the language, there's language barrier. So they say yes when they actually don't mean a yes in consenting. And then there's also dynamic and communal consent models and unified continental legislative frameworks. I can't move. Is it possible you move them for me? Okay, it has moved. So this is what I've talked about. Informed consent is very important when it comes to ethics, privacy and data protection, benefits of sharing, who benefits from the discoveries. If the samples are collected from Africa, who benefits from the discoveries? Fair collaboration and authorship, so that we avoid helicopter data. Community engagement and equity. Equity is another thing that is really important, ensuring African population benefits from precision medicine. Like, for example, what happened with the vaccine? It was a bit unfair because we could not get vaccine because we didn't have money to buy or we were not a priority. So we are hoping as we develop these things, we also get to the priority list. So avoiding genomic colonization is another factor. Exporting samples without local capacity is just like a colonization thing. And this has been happening for a very long time. And then limited African leadership in publication and grants and an equal global collaboration. I think I should have added something also on our governments are not investing in genomics. They're not investing in research. A few of the public institutions have funding. So most of us, we don't have funding for this. So opportunities for the students in the room. There is bioinformatics, research areas, genetic counseling. So we are working on developing genetic counseling training, computational biology, immunogenomics. Yeah, we are also, I think Dr. Karaoke is also doing something on HLA, and we really want to understand our HLA for the purpose of transplantation, that is renal transplantation, and also stem cells transplantation and any other transplantation. Cancer genomics, ethics, genomics research, people to be trained so that when you make decisions in the boardrooms, at least it will be informative, data science and AI, and public health genomics. And then building interdisciplinary skills is key. Learning, coding, and data analysis, collaborative internationally, collaborate with other international partners. We can't do it on our own. We need support. Stay curious and adaptable. Publish and communicate science. And when you are publishing and communicate science, don't forget those people that gave you the data. Let them also get to know what came out of the study. In that case, you are not saying your sample gave this. You say the samples that were collected in this community inform us of this and this. So that is, so these are a few initiatives that we are doing to try and bring this through immunogenomics. And last week on Friday, we had this expo by Moi University School of Engineering students. They invited me, and we are encouraging them to ask bold scientific questions, pursue interdisciplinary learning, build networks and collaborations, advocate for ethical research, and use science to solve African health challenges. The future of precision medicine in Africa will not be built elsewhere and imported. It will be built by African scientists, clinicians, counselors, bioinformatics, and innovators. Are we ready for that? Do we have them? So thank you all for the very long talk. I'm sorry for that. You can reach me by those.
Thank you very much, Dr. Cherore, and for sharing your insights on breast cancer genomics and also bringing a lot of ethics to this symposium today. So we'll receive questions and you can answer them from all of us. Any question?
Yes, thank you very much for this inspiring talk. And indeed, as you said in the very beginning, the diversity among African ethnicities genetically is very large. And I wonder in how far that it is an extreme challenge, actually, in doing personalized medicine. I come from the following idea. I mean, if you think of that the non-African humans, they perhaps have only the diversity of a single ethnicity here. And then most of the studies have been done on this single group. And from that, for example, the risk of breast cancer associated with certain mutation is derived. While on the African continent, you have so many ethnicities, and the risk for a certain mutation, given the different genetic background, may be different in all these ethnicities. So I really value and adore your efforts, but at the same time, it seems a really huge challenge. Could you perhaps comment on that?
It can scare when you really want to think about how diverse we are, and if actually this is going to make sense, is going to be economically good for us. Should we invest in this? But I'm really optimistic because, one, the technology is growing very fast. What was really expensive, I remember in 2020, we could do whole exome sequencing, and maybe if you are using nanopore technology, you could do 100,000 Kenya shillings. But now the sequencing cost is coming down because of technology. Same to bioinformatics pipelines. So I'm really optimistic, especially with AI, because AI can tell us, I believe it can make this process of individualizing analysis so that if you are matching somebody from Kenya, it has to be matched with somebody that is so close. So I'm hoping that AI will, and the technology will help us that. Otherwise, I agree with you. It's not as easy as it sounds. I've met several people, especially from outside Kenya, saying that why should you do these things? Yet people cannot even afford drugs that are targeting the mutations that you're talking about. But I'm happy that even those drugs are coming down. There's a lot that is coming from China. China kind of simplifies in technology, and many other people from India. And even in Kenya, they are innovators. We have very big potential. It's only that we are not investing in the right. So if we can invest in Kenya and have our own genomes, maybe for each and every group, then that will make a big difference. So, I agree.
Okay. There was a question.
Thank you, Dr. Rispa. So my question is on capacity building for bioinformatics. So given that the field is changing currently with the advent of AI and automation, how are we bridging the gap to make sure, because most of the trainings I've attended for bioinformatics is usually like scripting. These days you don't need to script, you just need to give like a Claude or GPT for you. So how are we bridging the gap to make sure that the bioinformaticians who are being trained right now not be obvious?
Thank you very much. I guess you're one of the students that we are really waiting upon to come up and take the role of being trained. So that's a very good question. AI is here to stay with us. And I'm happy that even at the university, we are now trying to bring AI into the curriculum. We have to. So I think even when training as a bioinformatician, there is a lot. So I think AI is being incorporated into many curricula. Yeah.
You have a question? Let's have one.
Thank you so much. What informed the decision to use zebrafish instead of, we've seen previously that for studies they use rodents. What informed the decision to use of organism? Thank you.
Thank you for that question. I wish the person that does zebrafish modeling was here, He'll be traveling in November. Maybe I should invite you so that you interact with him. He's at University of Linköping University, and he has spent most of his time from when he was a postdoc to research on this zebrafish model. And I was asking him the same question exactly, because I told him we have tilapia. Can I use tilapia larvae? And he said no. So this is a model that has been kind of made in such a way that and the uniqueness that it has. For example, it can allow cell-to-cell interaction. It is a transparent larvae. You can see the color changes, like you see where the tumor is and where it has gone. And the reason why they are moving from rodents, models from the other animals, is because there is a lot of interruption between the immune system of the rodent and what happens. So in this is a special model that can bypass that. With rodents, you use a lot of money. This is very easy. If you go to the aquarium in Linköping University, it's smaller than this room. But it has a lot of zebrafish models that can be even sold to other people. And you can kill most of them. Whenever they are working on it on a petri dish, the way you use the needle to inject the tumor, you kill so many of them. And if you were to kill rodents, so many of them in the process, you know how it will be costly. So it is cheap, and I think it mimics human immune system so well, and it can allow the transparent checking so that you can easily know if the tumor has spread or grown and so on. And I invite you to read more about this on BioReparia. There's a website on BioReparia, BioReparia, BioReparia website. Yeah. You read more about the zebrafish. I'm happy to have people that have experience in fish, because my feeling is that we should bring this technology. If it works for us, we should not be sending samples out because it's very expensive. We can keep the fish here and grow them here and do these experiments here. Yeah.
Okay. Thank you so much. My question or a comment, maybe clarification on that, is from the biochemistry perspective of it. Apart from just cancer, we have a number of infections or disorders, especially the diabetes and cardiovascular diseases, such as the heart attack, heart failure, which has really resulted to a number of death cases. So my question is, like, with the genomic and maybe from your way of seeing or from the way of thinking, is it possible for us to design or come up with a particular gene, especially which is so improved in a way that maybe through the bioediting way of it, so that maybe using, because from the biochemistry I just heard about the fish for the first time in terms of the biochemistry aspect of it, but for us we especially measure on oyster rats. When we want to do the experiments, the human-based ones, then we can now think from that view based on the results. So maybe from your ways, a way of coming with the gene, we improve the cloned one, so that maybe if there is a heart failure somewhere, maybe so that we can make more experiments on that and further advance in terms of the research.
You are Sharon? Yeah, thank you for your question. I can see that is a very inquisitive kind of a question, and I want to encourage you that yes, we can have unique genes. And how do we get these unique genes? We have to really sample many people. Maybe we do some population genetics, and then have the cases and controls, then come up with those genes that we are really sure that it is associated with the causation of the problem, and then work on those genes. So it's possible to have. I think from literature also there are genes that have been discovered around cardiovascular diseases like lung cancer, for example. There is a EGF that is doing a very good job at the moment. And Aga Khan are working with MTRH, and patients are getting better prognosis. We wish we could also get for heart patients with problems with the heart. But first of all, we really have, how do we know that this gene is going to work for us? We have to do a lot of studies for that, but it's possible. If we have passion and people who want to invest in that, we can find out which genes works for us or are associated with the problems that we're seeing. And then with that, now we can expand on developing technologies around that. Yeah.
Okay. I can see more questions, but we don't have the time. So I'll request all participants who still had questions. We are soon breaking for lunch. Please engage further. Dr. Torore, we are time bad. Sorry for all those that had raised their hands. Dr. Torore is my colleague in the School of Medicine. As you go and sit, maybe just answer one question I have. So for immigrants who've moved to maybe other continents, mainly Europe and USA, and of course from the slavery aspects, long time ago, is there relatable data showing then that they are also still at high risk for aggressive breast cancer, or what is out there compared to Africa? So as you prepare to answer, I don't know if the online participants are there. We would want to have Edith Kurui to prepare so that she can present for 20 minutes and then we can take a break. So maybe answer that.
Yeah, thank you, Dr. Tue. That's a very good question. So Olopade actually was working with the Black Americans in Chicago. And I think that is when now she discovered that there is something in the African people that is making them have aggressive forms of breast cancer. So yes, I think even in the representation of the genome, the small percentage is actually from the immigrants, not from Africa. But that is so small that is not informing. It's a very small percentage. I can't remember the percentage, but it's like 5%, not even 10. Yeah. So most of the 5% that is already now representing us is actually from the immigrants, either from Europe or from the States. Yes. Thank you.
Let's applause Dr. Torore in a round of claps. Thank you. Thank you very much. So we'll listen to Edith if she's ready for 20 minutes, and then we can take a break. Edith Kurui, are you ready? She's muted.
Hello, yes, I'm ready.
Okay. Let's see. Got the title. It's here. See. We will be presenting a talk titled Environmental Genomic Approaches for Surveillance of Fecal Oral Zoonotic Pathogens in Shared Water Resources, a One Health Perspective. Edith, karibu.
Okay, thank you so much. I don't know whether you can see my slide. Okay, so my name, I'm Edith Rui. I'll be presenting on environmental genomic approaches for surveillance of fecal oral pathogens in shared water resources, a One Health perspective. So my introduction is that globally we have a high number of people, approximately 2.2 billion people, who lack access to safely managed drinking water. And out of these 2.2 billion people, we have 15 million Kenyans are relying on contaminated. Yes, please. Okay. Is it better now? Okay. So can I continue? Okay, so in my study then I'm interested to—
Yes, can you hear me? You can hear me? Let's hope so. Thank you very much. Thank you. I just hope, my apologies for this break in transmission. I hope this time is going to be better. Can you hear me well now? Thank you. Thank you. Good afternoon, everyone, and welcome back. My introduction has already been done. So I started by talking about the story, what led me into the evolutionary genomics in One Health. I wanted to share a short story on how the journey started. In early 2018, Nigeria experienced one of its largest Lassa fever outbreak. We know that Lassa fever is endemic in Nigeria, but in 2018 we had the largest outbreak. Cases surged across multiple states, and healthcare workers became infected. And of course, we suspected nosocomial infection, but we were also very concerned because it was human-to-human transmission was not known to occur. And if that was the case, it would have been the implications were numerous, because sustained human-to-human transmission could signal the beginning of a far larger epidemic and regional and even global consequences. So then the question was Lassa virus now spreading efficiently from human to human? Yet traditional epidemiology alone could not resolve the growing uncertainty. Then came genomics. But we see by sequencing viral genome across infected regions, the scientists reconstructed the evolutionary relationship of the virus. And from there, they revealed that most infections were not driven by sustained human-to-human transmission, but by multiple independent spillover events. Then from there, what happened? This now changed the narrative of how this disease was being taken. So genomics separated fear from evidence. These findings reshaped our outbreak response strategy, strengthening surveillance, infection prevention, environmental monitoring, and rodent control, rather than focusing on widespread person-to-person containment. Of course, the 2018 Lassa outbreak became a powerful demonstration of the One Health concept. So we suspected that there could be various ways by which the pathogen is spread to human, because some of the persons had no history of contact with rodents. So today, evolutionary genomics allows us not only to detect pathogens, but to understand how they evolve, spread, adapt, and potentially ignite pandemics. In an increasingly interconnected world, integrating genomics into One Health surveillance systems is no longer optional. We all understand that, even from the previous presentations, that we have to integrate One Health surveillance system for health security, food security, and even because we live in an interconnected system. It is essential for effective prevention and outbreak response and pandemic preparedness. This now takes us to From there, the thirst for disease surveillance became huge because we now know that we now took it as our frontline defense against infectious diseases, which caused profound loss, particularly of lives, particularly in Africa, suffering from war, malnutrition, and of course, some of our cultural behavior that also leads us to this, and our biodiversity. So this caused a lot of economic loss as well as loss of life. We all know, and we have heard it over and over, that about 75% of emerging infectious diseases are zoonotic. This is just because we live in the same environment and share one ecosystem where we have human and animals and the environment interconnected. So dealing with a pathogen means we need to to take into account the human, animals, and environment, because the health of one is the health of all. So zoonotic reservoirs are where pathogens undergo genetic evolution that enhances their host adaptability, transmissibility, and virulence. This is where it is silent or noticed. But because we encroach mostly in the animal environment, and of course for food and for habitation and even for exploitation or for economic reason, we always encroach in their environment. Therefore, we are prone to, and we share this environment with them, and therefore we are prone to infections by a genetic pathogen. So pathogen of evolutionary genomics, which exploit genetic variation and the evolutionary pattern of organisms, has become an indispensable tool for understanding how pathogens adapt, spread, and evolve before, during, and after they cause disease. These insights have been harnessed and to inform transmission, pathogen characterization, and then diagnostic development, vaccine development, and control and preventive measure, because they help inform public health policy for health security. Having said this, I want to start with the first journey. So after the 2018 Lassa outbreak, we embark on two major studies in order to using evolutionary genomics to expand our understanding of Lassa virus transmission dynamics. This was between 2018 and 2022. So the first, from the two studies, we detected using PCR an exceptionally high Lassa fever prevalence in small mammals, that is rodents, that is mice and screw, screw, and then rats. And the prevalence was 61.6% using PCR. This was the first time we were detecting this, because in the past, Lassa detection in rodents, the maximum was 10% in various countries where Lassa is endemic. So it became very difficult to understand that what is happening if the prevalence in rodents is this low. Why is it that we then have this low prevalence? But we use a different approach because we use the organs of the animal, not blood. So this enables us to find this high percentage positivity in the Lassa virus in small mammals. There was also, apart from that, we also discovered numerous small mammals. We discovered that all the small mammals, rather than Mastomys matalense, are infected with Lassa because they share the same ecosystem. There was also evidence of active and repeated infections from this small magma because of the IgM dominance and IgM co-detection. This shows that Lassa virus was being constantly shared and reinfecting animals as they move, small rodents. So we saw both the same animals, we could measure both Normally, immunologically, once IgM is detected, IgG is detected, you no longer find IgM. So Lassa virus, as we continue our zoonotic surveillance in understanding the transmission, it then became important for us to sample non-small rodent mammal. Then what we found was not surprising to us because we know that they share ecosystem, the same ecosystem, they eat in the same environment, with rodents. So Lassa was detected in multiple non-rodent animals, dog, pigs, cattle, sheep, and lizard. So this then we also changed the narrative because once we control rodents, we were thinking that Lassa, even if we finish all the rodents, then Lassa would still be in the environment as well as in a non-rodent animal and then co-infect. More importantly, the infected animals appear healthy, so they could be a silent reservoir. But future study will enable us to understand the transmissibility of the live virus shared from those animals. This is still work in progress. Well, now using the next generation sequencing, I'm sorry, my slide moved. Next generation sequencing, we were able to assemble 53 new Lassa virus genome, the largest rodent data set today, and 6 Lassa virus genome from non-rodent animal. What did we do with it? Looking at the downstream analysis and the phylogenetic analysis shows that there was cross-species transmission as the rodent data as well as human and other animal clustered together. So it shows that there is a cross-transmission between species, human and animal. Then looking at the genetic diversity, we observe that it is maintained by the exotic persistence in small reservoir. Why? Because sequencing data demonstrated that a human clinical sample from specific state will share exceptionally high sequence identity with a rodent sample from the same state and even sample collected decades apart. This shows this lack of linear temporal accumulation across region, but shows that the virus is usually locked in this localized environment or localized geographic region. That's why we have different lineages in different parts of the country. So having done that, we accidentally detected some lineage 2G in Ebonyi State, which is one of the states where 400 kilometers apart from where we see lineage 2G for the first time, showing that the virus can also move with us or move with other domestic animals into another region where we usually know that is locked, but yet we could find at least one there moving. So also we move that virus is also moving. There was also strong evidence of interstate movement of the virus between the two states, southwestern states. So this slide shows the prevalence of Lassa in small mammals that we've described now. That is rodents. We usually call them rodents, but because of shrew, I call it here small mammals. This was when we sample only looking at the rodents. So we see the high prevalence, and then the second figure here shows the prevalence in non-rodents in different species of animals, and then it varied. So some animals carry more virus, carry the virus more than others, and this work was done over 4 years of sampling. And then the phylogenetic analysis shows of lineage 2G of Lassa virus sequence here, showing cluster with the human and rodent strain. This is demonstrating the cross-species transmission we talked about just now. What this means is that what we have to retain from this is that while rodents, that is small mammals, are still the major reservoirs, domestic animals can silently spread the virus because they cluster together. And then sequencing cluster by geographic and not by host species, suggesting cross-transmission. Then we also use evolutionary genomics in to address some of the food security diseases, particularly African swine fever in Nigeria. So what did we do? After an outbreak of African swine fever in southern states in Nigeria, we decided to do a surveillance over after the response to that outbreak over in many states. With next generation sequences, we assembled 27 African swine fever genome, the largest number of genomic data set in Africa to date. Then what did it tell us? Of course, downstream analysis saw that all the sequences were for genotype 2. That is Georgia strain genotype. Yet the question remains, is it the same globally circulating genotype 2 strain variant, or is it a different one? What did we see? Looking at the variant, we observe numerous single nucleotide polymorphism count, more than 200 in the African swine genome collected in Nigeria, mostly affecting the multi-gene family. These genes are involved in the virulence and also as well as the immunity, host immunity. This mutation, and then we also saw recorded missense mutation in the CD2V and the P22 gene. What that tells us, because CG and 22 play important role in viral virulence as well, and host interaction and the transmission biology. And then these are important targets too for evolutionary analysis. And sometimes they are also used for vaccine design and molecular epidemiology. Because they are used for vaccine design, it became very important for us to list it here. Now, what happened? Then for some structural variation, we recorded large deletions and then of LRV gene 1 exclusively in one state, and then LRV2 gene in Osun and Ogun State, and then the RVV gene in Ogun State. But there was no recombination detected among the samples. Then went into phylogenetic analysis. The phylogeny shows a global geographic structuring, meaning that we had some Nigerian clades diverged significantly from other clades, but most related to the West African clades, the Nigerian clade here. This shows that in considering a vaccine or drug development, there is no one size fits all, as we heard today. So these clades also differ with the subclade from East Africa as well as South Africa and then the Euro-Asians. So they were different clades here in green, orange, blue, and then red. That regional, this shows a regional transmission pattern that needs to be taken into account when designing drugs or even vaccine, because there's region-specific viral evolution and adaptation. The next food security disease, we tried to use evolutionary genomics to survey that I decided to select today for this presentation is the lumping skin disease virus. This is a disease that affects cattle, most especially, particularly seen because it's one of the transboundary animal diseases that are of great economic importance, and it's caused by Capripox virus. And these animals affect the skin and, of course, create a debilitating condition where there is loss of productivity, and then milk production being the and skin becomes apart from on thrifting it, there's we cannot use the skin of the animal because of the lumps on it. So what we're using next day, we collected samples from 3 countries, Nigeria, Cameroon, and Benin, because they are transboundary, and then animals come from those countries into Nigeria. And of course, we needed to know where they come from. So what we did was we sequenced samples collected from those countries and recorded the highest positivity. I mean, we did PCR and recorded the highest positivity in Cameroon. And genomic analysis suggested that there's a distinct West and Central Africa sublineage of the LSD virus, showing that there was no large difference. So here we see the phylogenetic tree of the LSD, showing a strong cluster between from West and Central Africa from this with this lineage 2 and 1.2, suggesting the emergence of a new regional sublineage, which differ from other lineages from South Africa, Middle East, and West Central, Western Central Africa. So overall, these mutations were linked to immune evasion and virulence, raising concern about vaccine effectiveness as well. This was almost similar to what we recorded with African swine fever virus. Overall, our study highlighted that LSD in West and Central Africa is evolving as a distinct regional lineage with mutation that may compromise current vaccine and alter transmission dynamic. Again, another area where evolutionary genomics was used to explore the hidden ecology of wildlife genetic pathogen. Of course, I am only focusing here on the evolutionary genomic aspect of the work. There were questionnaire, we look at the value chain and the rest of, and others, and even PCR. But what is of interest to us here today with this topic is the evolutionary genomic aspect of it. Using a next generation sequencing of some selected pathogen from the samples we collected, particularly in Lagos, that is economic city in Nigeria, because bushmeat are moved from that to various countries from that site and also come from neighboring countries like Benin Republic into Nigeria to ship out of the country because there's market. What did we find? We found some important zoonotic pathogens such as hepatitis E virus, particularly the genotype 3 that was detected in warthog, and of course the gementic virus that was detected in an antelope. These are viruses that are very the gementic virus is it's also new in Nigeria. It has never been reported, but we saw that this gementic virus sequence cluster with the African lineage. I mean, that has already been reported in other countries. However, the hepatitis E virus genotype 3 sequence was very divergent, but clustered with human-associated strain, highlighting zoonotic potential and hidden viral diversity. So the validity analysis of this then now links to strain from Africa, European, and Asian lineage. This suggested viral recombination, but we need to still do the recombination analysis to see where the recombinations are to be able to inform public health. And of course, there is global viral connectivity as this cluster with other viruses from other in the gene bank that are from other locations. These are our findings. In conclusion, our findings highlight that wildlife represent a major and undersampled reservoir of genetically diverse zoonotic virus. We then move a little bit, I'll move a little bit from here to bacteria. So that, oh, my slide is frozen. I don't know why. Okay. Where did we use evolutionary genomic again was for Mycobacterium tuberculosis complex surveillance. This is because this work was done because we know tuberculosis is endemic, and we've been suspecting that And there's a positive of genomic data out there, really telling us how, if using this One Health approach, we have this TB, the same TB we have in human, is the same we have in animals. So we were trying to understand why we have a lot of TB. Is it just from because it's airborne, or is it because we are getting it from other sources? So the first part of the work, which I'm going to share with you here, is the sequencing of some of the lung tissue collected from the abattoir in the northern part of Nigeria. The lung tissue showed numerous needles, so we picked it rather than using the conventional method of culturing. We went straight to using next generation sequencing. What we did was we used integrated metagenomic and metatranscriptomic analysis to understand whether there was really active infection of whatever microbacterium we will have seen. I forgot to mention here that this another colleague cultured this pathogen of course, and this did the culture of this sample, and then of course confirmed that it was Mycobacterium tuberculosis complex. But then we also found that it was with the sequencing, we were able to assemble the genome from all the samples to be Mycobacterium tuberculosis complex, all the 10 sample sequence. And then, okay, there was also a strong RNA activity, which confirmed active infections, not just the persistence of DNA, but active infection, because RNA genes that they were expressed, given that this is a bacterium. Then with the standard genomic method alone, we suspected that, we found that it was M. bovi. But with more detailed analysis using some of the very important marker, the RD4, RD9, and ID12, which are very vital molecular markers for identifying and classifying bacteria within the Mycobacterium complex. We use that to be able to separate, whether to know whether it was Mycobacterium bovis or Mycobacterium tuberculosis. So using those, particularly the RD12, enable us to classify it as being the Mycobacterium tuberculosis because of the deletion of this gene. So once these genes are absent, it shows that is tuberculosis, M. tuberculosis. So this shows that single method approach are insufficient for accurate surveillance, and of course, evolutionary genomics based methods such as phylogenetic now help to achieve better characterization of the bacterium. Next, we also harness these trends in genomic sequencing, as well as and the downstream analysis to look at the antimicrobial resistant gene in transboundary livestock system in Nigeria. So this was still the cattle in the northern part of Nigeria, that is Kano, Sokoto, and Adamawa, in 3 states in the northern part of Nigeria. So we also went straight using swab samples collected from those animals, particularly animals from the abattoir, and then cattle market and some farms. So the sequencing data shows us relative abundance of antimicrobial resistant genes classes. They were very, very abundant. This revealed that we have a lot of antibiotics from different antimicrobial pathogens. We've heard one of our participants presenting some of the antimicrobial agent which they found antimicrobial resistant gene. But with the power of genomics, we were able to see that grossly, and then numerous that we could tell that we expected than what we expected. But we use 2 methods of analysis, the read-based and then content-based rhizostome profiling approach. We were able to see that there was in this concordance, this overlapping of detection of antimicrobial resistance by the two methods. This strengthened the confidence in AMR detection and, of course, improve the reliability of genomic surveillance because using one method, we saw the same somewhere, and of course, different, and of course, they were also complementary because using only one method, we were missing the rest. There was also co-accessory resistance structure illustrating the prevalence of antimicrobial resistant gene. This had helped us to distinguish the widespread endemic antimicrobial gene from newly emerging niche resistant determinants supporting targeted prevention strategy. And again, we also saw that there was antimicrobial resistant plasmid co-localization network in this in the last one. What does it mean? It means that there was this. It shows this potential horizontal gene transfer pathway that the plasmid will help us transmit antimicrobial resistance from bacteria vertically. And this is a risk for rapid dissemination of resistant gene across bacteria population and environment. So we saw that we concluded that resistome is diverse, clinically relevant, and mobile. Okay. Next, this is just we've concluded this. We said that the core resistome we found in more than 80% of samples were from multiple antibiotics, particularly the tetracycline. Of course, others were there, but we focus on these ones that were occur in more than 80% of the 46 sample sequence: the tetracycline, the beta-lactam, the aminoglycoside, the polymyxin, and then bactericin. On several notable genes were detected at moderate and low prevalence level, including clinically significant genes and genes commonly associated with mobile genetic element. The one thing that was also striking is that we saw these genes, particularly the aminoglycoside modifying enzyme gene, and then the sulfonamide trimethoprim, which is just some of the, and the quinolones, which are some of the rare antibiotics that we use that already have a lot of resistant gene among cattle population. Very, very, very striking. It's still emphasizing the One Health, importance of the One Health approach in AMR surveillance. Then genomic characterization of a microbial antigen-resistant gene also concluded shape AMR diagnosis. How? Because we sample animals from farm, livestock, and abattoir, we were able to see this difference in abundance, particularly in the abattoir sample. Multiple and numerous antimicrobial resistant genes from sample from the abattoir. And this differential abundance is very significant in the sense that it shows that how very important it is, because we consume this meat. There's no time for the antibiotic to leave antimicrobial resistant gene to leave the meat before consumption. So we are taking it indirectly. So different management practice shape distinct AMR profile, as we see with the first image here. But looking at the microbial taxa, we also observe that there certain microbial community may promote or suppress AMR dissemination. This shows that there's a potential for microbial indication or intervention target for AMR control, because some of them may even just promote or some may suppress AMR, some of the bacteria. Then in the third graph, this shows the positive association between antibiotic use this was said when this reinforces the need for regulated antibiotic use and the stewardship policies for antibiotic use, particularly in the livestock as well, because we may take it from there and even human. This is particularly important because these animals are just found in the market and then in the abattoir. So it becomes very important to control such. And then of the fourth, the D figure G shows where all of these animals come into Nigeria, then Niger, Mali, Cameroon, and Benin Republic. This regional AMR connectivity access trade. Livestock movement contribute to regional dissemination of resistant organism and antimicrobial resistant gene. This then highlights the need for a coordinated cross-regional surveillance and biosecurity, of course, and then the One Health intervention. I will then move on from bacteria and AMR back into viruses. It became very important for us, knowing that we are often threatened by epidemic and outbreaks in our region, because Nigeria lies between that region where there is substantial number of undocumented viruses and zoonotic, making it a priority area for surveillance. Of course, there's a lot that has been done, but we are always surprised by what happens, and we seem not to be learning. So it's very, very important that disease surveillance that is untargeted, not targeted for pathogen like we did for Lassa, African swine fever, and the rest, and other pathogen, yellow fever, or we will need to use One Health genomic surveillance to be able to prepare before this gets to us. What we did through a One Health genomic surveillance program, we embark on sampling human, animal, and environmental sample from 2 states in Nigeria, southwest and southeastern states, previously impacted by Lassa. We started with the animal sequencing, where we use metagenomic next generation sequencing for 240 individual animal samples, so representing 11 distinct taxa. So that is bat, cat, chicken, dog, goat, lizard, pigs. All these animals are in contact with human. So the idea was to see what is circulating within the communities in those states to be able to inform public health. This is just an initial stage of the work. And of course, work is still ongoing and analysis as well. So what we observe on this figure is that we have intersection between the pathogen we have across all the sample we identify. Let me say, first of all, that using next generation, we had 214 distinct viral taxa, spanning 33 families, viral family, of 1% represents novel species by ICT criteria. That is using the reference of ICT reference of 2022, showing that we have a lot that we don't know about and circulating among animals. Here we see that some pathogens are found between animals. And then the number of animals where we find it. And then some of them, we see a pathogen's broad host adaptability, increased epidemic and pandemic risk. Ultimately, this will enable us early detection of emerging variants before widespread transmission occur. And that is the ultimate goal of our disease surveillance, to be able to track it before it gets for countermeasure preparation. I try to show some of the family, many family, and also some important families that we see, and of course known already to cause epidemic and pandemic in human, and also viruses of economic importance in animals, particularly the paramyxoviridae and then the coronavirus, the RNA viridae, which is where Lassa fever virus come from, and then the nairoviridae, where we have Crimean Congo hemorrhagic fever, the pox viridae, where we also have some mpox and the rest. So this shows that we see this variety of, although some of them were not able to assemble the full genome because it's metagenomic with all his, with some of his weak points because of competition in terms of analysis is not targeted. But what we have here is that later on, maybe we'll do a targeted sequencing of some of these viruses. Now, well, I am going to pick some of the families here and then pick some of the important and novel viruses that we saw that could be a potential threat during our genomic sequencing. I will first of all start with the corona. I want to say, is it coronavirus now? No, I will start with the piconaviridae. First of all, the piconaviridae family. That is where we have many of the high priority viruses. These are viruses that WHO has called priority virus, important virus, viruses of priority that causes enteritis, gastroenteritis in human. So this, and then most of them belong to the piconaviridae. Here we found the red dots here at the tip of our tree are the samples with those viruses that we detected during our metagenomic sequencing. We have here the hepatitis A, the ecovirus, the enterovirus, the Succini virus, the Megri virus, and the Epasi virus and the parechovirus. These are all high priority virus. Why are they important? Why we've seen it here, and then for them clustering closely with human-derived Nigerian and Cameroonian sequences, as we have here, is not clear here because I didn't see the. I forgot to mention to put the legend of the black dot, but this only shows that there's this. There's a possibility of animal-associated virus closely related to human, suggesting the potential spillover at the human-animal interface. And these samples were mainly swab samples, oral and rectal swab samples from animal. And given that they are oral and swab samples, it means that there's increased risk of future outbreak from this pathogen. So these are pathogens we need to look closely at, and there are very many and from many various animal species. We have there in dog, chicken, goat, pigs from the samples we sequence. Next is I'll take a hepatitis A virus that we found in goats. This is just the same figure we just looked at now, but because they are still between piconaviridae, we pick the hepatitis A virus from there and trying to do this phylogeny. So this hepatitis A from goats cluster closely with the Nigerian human genotype 1B, including the isolate from Lassa fever patient where it was first recorded. So this shows that finding it in goats means that this viral reservoir is still in animals, and then showing the importance again of using genomic evolution and of course in One Health. I'll move to another novel lineage in nairoviridae. In nairoviridae, we see the Crimean Congo hemorrhagic fever, which is a viral hemorrhagic virus that also cause outbreak in human. So with large animal, particularly cattle, and it's tick transmitted, known to be sometimes the reservoir because there's increased seroprevalence of Crimean Congo a hemorrhagic fever in the northern part of Nigeria. But these samples were collected in the southwest and southeastern part of Nigeria. But what we see that is a novel lineage called the Chiophora lineage. And this lineage was first described identified in Screw in Senegal. But this was also found in Screw here in the southeastern part of Nigeria, showing that forming this distinct relation with that, the ones that were reported from Senegal. This again shows that viruses, in looking at genomic evolution, we could find some of the viruses may mean, and then that are known to be, that were reported far away in another country. This is important because we do not know how they will evolve and move into human, and of course spread in this region. Next is the Arena viridae. I cannot live without talking about Lassa. We saw numerous Lassa. These are the yellow ones, are the ones from this study, where it is put IDH here. We also detected numerous lineage 2 Lassa virus related to the previous outbreak in Nigeria, meaning that these pathogens are very much around from various hosts, are very much around in our locality, be in Edo and Ondo. The question now is, with this virus, can we design vaccine that is Lassa? There's also a vaccine development already there. But from what we've seen here, there are some strong mutations which I'm not able to show here in the course of, in particular in NP and GPC gene that are selected for vaccine development, meaning that we have to continue our surveillance to see if this vaccine that we are designing will still work when time comes for the trial and for the implementation. That is the importance of this for now. And then one important animal disease selected here is from the Paramyxoviridae family. That is the peste de petit ruminant virus that causes a lot of economic loss in the small animal industry. And then the WHO has, and the EAO has planned the eradication plan in 2030. So what we detected, the 2 full genome assembled during this project, we cluster with the lineage 4 PPR detected in goat from Ebonyi State. From Plateau State, in 2020, suggesting an ongoing rotavirus circulation and diversification. And the phylogenetic dispersion indicate multiple transmission events rather than a single outbreak source, and continue endemic evolution of PPR within Nigeria. This disease spread all over Africa and affects the small animal industry significantly. And then with this, the question is, with the eradication plan, we will need to have a lot of data from this PPR virus to be able to plan, to be able to eradicate this disease. So the next, here, I will want to finish by just talking about some of the key takeaways is that metagenomics detect emerging threats early. We have noticed this, and then pathogen evolve across interconnected ecosystem. That's why we are advocating, and we keep advocating, the integrated One Health system at various levels. And then, although we are focusing here in this pathogen, but of course, the host interaction is also an important thing that we should be looking at how we respond. But the countermeasures are usually work across. Then evolutionary genomics inform knowledge of variant transmission, variants transmission, vaccine design, and of course inform policy for public health action and prevention across this interconnected ecosystem. The implication of evolutionary genomics within the One Health framework is essential for designing and informing several preventive and control measures. Yes, this is not news again, and it's sort of what we've been saying from the beginning. And then evolutionary genomic trace transboundary movement and spread of AMI regionally, as I've tried to show in the course of this presentation. And then in Africa and beyond, One Health genomic surveillance system will help identify pattern of antibiotic resistance, track mutation in pathogens, detect emerging pathogen of public health importance, and improve regional readiness for pandemic. In short, investing in global disease monitoring through One Health genomics is an investment in health security and stability, knowing fully well that infectious diseases are the ones ravaging the most human population and even fast. So it will be a good investment for us in Africa, and not only seeking for funds elsewhere. And it's important for us in Africa to start looking into investing in this disease monitoring, to put a strong surveillance system in place. What happened in the DRC recently, it shows that we are still not prepared. And then because of the war and the limited infrastructure and fund in that regard, we are still lacking. And then this keep coming, and then it's high time we invest in this, and then be very, very intentional about investing in health security and stability in the sub-region. And what are the current challenges? Many have been listed, and then the emerging opportunity, many have been listed. So it will be a sort of repetition for me to start again. We did talk of limited, integrated, coordinated genomic surveillance system. We have limited sequencing infrastructure. We know it is the power to put data there for countermeasures development and even public health policy. Of course, we also have some limitation in bioinformatics and data integration challenges and data integration. And we do see the emerging opportunity is the AI and system biology, for which offer transformative opportunity. With that, I want to thank our director, Professor Christian Happy, for putting up the platform which we work for our One Health genetic surveillance. The IGH Invita team, many of my students here, and that also they conducted this study, not me. I only guided. And then the entire IGH community, some of our collaborators and partners, the NVRI, that the Nigeria Veterinary Research Institute, the Nigeria Center for Disease Control, and some of our collaborators from Cambridge, Florida University, Harvard University, Washington University in Europe. Also thank many of our funders, many to be enumerated, who funds various projects. I also want to thank those who offer their animals and then to be sampled for this surveillance. Thank you all for listening.
Professor Anise, we followed you very well. Now we'll welcome.
Thank God.
Yeah. We'll welcome a few questions, and you can be able to respond to them. So we welcome any question.
Thank you, Prof. Anise, for that wonderful talk. I have a curious question here. I know there is a lot that has led to discovery of these novel variants. I don't know what that means. Is there anything we can do to kind of maybe develop vaccines just to prevent these variants, trying to be ahead of the problem that we are seeing? I don't know what you can advise or comment on how to actually deal with these variants. Some of them may be causing resistance, as we've seen. So what is it that we should do to have that impact? can it inform vaccine development, for example, or change of treatment? I don't know if it goes with the kind of treatment that we have. Yeah, because it is really interesting to discover these variants, but what impact can we get from these discoveries? Thank you.
Can I respond? Okay. Thank you very much, Professor Rispa. I think as we said, as I said during the presentation, this variant is very important because it informs public health on what to do. And of course, for vaccine development and even diagnostic development and development of every other countermeasures, it is important to know what are the variants, because if the variation is at the site where the vaccine is targeting, of course, if you have a vaccine that cannot target that very one because it has mutated, of course, it will not protect. And in terms of diagnosis, if there is a variant that has affected diagnostic tools we use on ground, it therefore means we will have a lot of false negative. It means that we will not be able to control. So these variants, these new variants enable us to design, this genomic data enable us to design new vaccines and of course even new diagnostic tools for control and treatment and even prevention, prevention, control, and even treatment most of the time, because we need this information, genomic information, because drugs are designed based on the pathogen itself. And if then there's variation, we need to move with it as well. Thank you very much. I hope I was able to address your question.
Any other?
Hello, this is Olivia from Germany. Thank you for your presentation. I have a question about your Lassa virus survey. You mentioned there was evidence of interstate movement. Can you?
Yeah, for Lassa movement, we use the Macau jump analysis to see, apart from the clustering we saw in the phylogeny, we wanted to know, using the evolutionary method of analysis, that is the Macau jump analysis. I didn't present that. I didn't present that slide here because it makes it a lot. There was this interstate transmission of the virus across both states. And that is, they are very close states anyway, the Ondo and Edo states. So showing that the virus move across those states. And of course, in the past, we had the Lassa fever center in those states. That is the Irua Specialist Hospital, where we had most of the human cases. And patients used to move from a patient diagnosed for Lassa used to go to Edo for treatment. So, and now after some time, there was the reverse. So this was also shown during our with our genomic sequence analysis, that it shows a lot of, we had a lot of cross moving of the virus across in both states. Thank you. Over.
In phylogenetics, when do you classify a variant as novel? Because according to me, when it has been reported by one country, then the next country will report it as a variant that was seen and no longer a novel, just generally, because we all talk of novel variants. So how do you classify a variant as novel in phylogenetics?
Well, you mean the Theophora from the narrow Viridae family, I guess.
And you are seeing in your study population. So I don't know
Okay.
what mechanisms you use so that you classify a variant as novel.
Okay. I thank you very much for that question. I think I want to mention that using the genomic data, we we detected one of the person, the Theophora virus in shrew. This was reported, but they vary differently. That shows that they had more than 50% acid, more than 80% amino acid difference using some very important marker. That is, this was a RDRP gene that differ from the one that was reported. So it's a novel in the sense that using the criteria I mentioned, the ICT criteria that I mentioned earlier, that usually when we have more than 20% difference, 20% difference in a particular pathogen, then of course that is shown to be known by ICTV criteria. So that was why it was novel, although they have reported, and this is a novel variant because that has not been, is similar because they fall in the same, they cluster in the same sublineage, but it was very different. It was very divergent from the one we know that was reported in Senegal. That is why it is, to me, is novel. We also found the one we talk about is the coronavirus. Another one was I don't think I mentioned it here, the coronavirus. We saw the rodent coronavirus, that is the IB, infectious bronchitis, very similar to the infectious bronchitis virus that we find in chicken. But the ones we found in the rodents here were very divergent. I think I should pull out my slide again. Where do I have it? I don't know where it is. That's where the novelty comes from. The novelty comes from the variation using the nucleoprotein and the amino acid and then the nucleotide difference to determine a variant novel because because they had a different, they were very divergent. Over. Trying to trace it.
And I think those were the questions. Thank you again for sharing your insights on the role of evolutionary genomics in zoonotic pathogens and disease and their use in surveillance. So thank you again and welcome to Eldoret next time.
Thank you so much. Thank you.
Thank you. Okay. So we'll proceed to our next presenters who are with us here. We still have 3 presenters. So we'll begin, we'll proceed to Olivia Dimhoff from Germany. And our presentation, you could be loading the presentation for Olivia. It will be entitled Wildlife Recovery and Disease Surveillance in Changing Reforested Areas Using Metagenomics. Welcome, Olivia. Welcome, you can use that.
Thank you very much.
Today I want to present the topic of my PhD studies that is investigating the impacts of restoration on biodiversity and emerging zoonotic disease dynamics in the Afrotropics. I'm a veterinarian and second-year PhD student from the Helmholtz Institute for One Health, short HIO, from the north of Germany, and the city is called Greifswald. And I work in the Evolutionary Community Ecology Group of Dr. Jan Gogarten. The institute was founded 5 years ago, so we are a relatively young institute that is still growing, and this is our current team. So we're about maybe 100 people in total now. But I arrived here to this conference from Kibale National Park in western Uganda, where I reside since September 25 as part of my field studies. So my PhD work is part of a greater project called Restorate, which is short for restoring ecosystems to stop the threat of re-emerging zoonotic infectious diseases. And we have overall 16 partner institutes and universities and operate in 14 field sites. I hope you can see that. Yeah. In temperate and tropical forests and wetlands. And my work concentrates in the tropical forests in Côte d'Ivoire and in Uganda. The central idea of the study builds on the hypothesis that restoring former degraded land with restoring the former degraded land, wildlife species would return to this land and biodiversity increases. So with greater biodiversity, especially for non-competent hosts for certain pathogens, the risk of spillovers of zoonotic diseases from animals to humans would therefore be lowered, which we would call the dilution effect. However, before forest structures and forest communities can be built for the dilution effect to be there, it can be that first the risk increases, the risk of spillovers, which we would call the amplification effect up to a certain point. And then we hope that the biodiversity in forest structure dilutes the pathogen. So that's the idea. And to measure the restoration gradient, we use the space for time approach and chose our sampling points in areas where restoration effort was initiated in the same year of sampling. 5 years after restoration effort was initiated, 7 years, and then 10, 30, and over 35 years. This is the example of Ivory Coast. And the last one would represent mature forests. So our study sites in Côte d'Ivoire are the 2023 built Cavalli Nature Reserve and Thai National Park, where we sampled from February till June 2025. We chose 4 sampling points in Thai National Park, 2 in the primary forest, an area that's been protected since the 1980s, and 2 in the secondary forest, an area you can see here, the lighter green, that has been annotated in 1995. And in the nature reserve, We chose plots like restoration plots where restoration effort was initiated in 2016, 18, 2020, and 25. In Kibale National Park, sampling follows a different pattern. There we sampled 9 sites with different restoration age from 5 to 75 years, plus 3 plots representing mature forest in the north, so here, in the southern parts of the national park. We sample areas that previously have been logged, which you can see here in black, and active planting compartments, which would be in the southern part of the forest. And sampling is occurring alongside traditional biodiversity monitoring on a monthly base for 12 consecutive months to also capture seasonal dynamics. So we're doing this from September last year till September this year. And we use eDNA, which is short for environmental DNA, that animals live in the environment in form of leaf swabs and carrion flies. There are vertebrate communities in our study areas, but we also use the same carrion flies and additionally mosquitoes to screen for bacteria and viruses in those areas. And all 3 sample sites will be processed and analyzed using metagenomic next generation sequencing approaches. So for the flies, we trap flies in each of the restoration plots each month using these self-made triangular nets, following by storing them in silica to completely dry them out until the analysis. And the study of my PhD fellow found out that you can process a large number of flies. So here this is the process. What she did is she was comparing a destructive protocol, which we used before, for extraction by a non-destructive one. And she found out that with the non-destructive one, you can actually process a large amount or a larger amount of flies, which would facilitate the process also of covering large areas with the eDNA. And then for the leaf swabs, the work of my colleague Christina Lingard also found out through swabbing leaves for a certain amount of time to collect DNA from the environment, we can create a picture of what biodiversity looks like in those areas. And just explaining the diagram of a former study. So we, in this study that we published, we took 24 swabs. Each swab was sampled for 3 minutes, so also relatively short time. And in total we could detect 52 genera, of which, with this is not shown here, 30 to the species level. So also a relatively high amount to the species level that is also higher than any other terrestrial eDNA detection methods. And also with a single swab or 2 swabs that could detect up to 12 genera. And to analyze samples from both flies and leaf swabs, we will amplify the samples targeting mammal primers, which is then followed by a verification step via gel electrophoresis, and then library preparation and Illumina sequencing. And for the analysis, we created an OBTools pipeline for each of the different sampling materials we use. the carrion flies can also be used as rapid tools for pathogen detection, which could be shown in previous studies of our group in Thai National Park. So Thai National Park is known to have regular outbreaks of anthrax, and our research could show that about 8% of the flies that we captured were positive for the bacteria causing it. So this is the map of the previous studies where we compared mature forest to the forest edge to the nearby villages, and the yellow is positive for anthrax causing bacteria and the blue is negative. So you can see in the mature forest there's prevalence of anthrax, but here not so much. And my PhD colleague Benito from Cote d'Ivoire is currently investigating the flies that we caught last year. And we could already successfully show that, or successfully detect anthrax in the mature forest, but also in the secondary forest I showed you, and in the restoration sites outside the national park. And we were also able to detect another viral pathogen that is known to occur in the area that we are investigating at the moment. So it shows that the flies can be used as rapid tools for now DNA-based pathogens. Another layer we added that I'm investigating is how mosquito communities and the viruses they carry differ in those different restoration sites. For this, I'm establishing an all-in-one metabarcoding protocol, where we first barcode the different species to build a reference data bank that we can use for metabarcoding also. For this study, but also for future studies that will come. And then we create mosquito pools, and we screen those pools for different arbovirus families. So for flaviviruses, we also use the metabarcoding approach, whereas for alpha viruses and Ottobonia viruses, we will still use qPCRs to detect. And as a last step, we will take blood fat specimen to see and analyze those to see the host diversity to add an extra layer of diversity to the eDNA biodiversity layer. Yeah, so mosquitoes are trapped using CO2-baited BG sentinel traps, then followed by morphological identification on site or in the field, and then pooling by month, restoration site, and species, and then the blood fat would be extra. And then in the laboratory, that is not in Kibale National Park, but Queen Elizabeth National Park, we would pool the mosquitoes further to for molecular analysis, so of the barcoding and pathogen screening. And the idea is also to build this reference library of mosquito sequences for future studies. The sequence that we create will be accompanied by pictures we take in the field, showing distinct features of the mosquitoes that hopefully a lot of people would use for their studies. And although we're still in the sampling process in Uganda and at the moment analyzing the data from Cote d'Ivoire, I wanted to show some really preliminary results of the general abundance detected in the first month in Kibale National Park only based on morphological identification, which here shown the relative abundance, and each of them is a restoration site. So you have, I don't know, probably cannot read it, but like here's 100 years, then going until 5 years. And we also added for the mosquito trapping a 10th site, which is the village, which would serve as a reference point of degradation. And it's no surprise that we are trapping in the forest, so most of the mosquito species you find are forest dwellers. But you can already see that each of these different plots harbors a different diversity. So it will be very exciting to see once sampling and analysis will be completed of this 12 months. Yeah, and together with the biodiversity assessment via flies and leaf swabs, and the pathogen detected by flies, the study will create a multi-layered health assessment of the environment in these afrotropical areas that hopefully inform future conservation efforts, but also help facilitate rapid disease surveillance. And with this, I'm already at the end of my presentation, and I want to thank my peers and my supervisors and my collaborators in Germany, Cote d'Ivoire, and in Uganda. But also I wanted to say special thanks to the field assistants, here my field assistants in Uganda, that collect these vast amounts of samples every month, and I think without them it would not be possible to do such studies. Okay, so thank you. And here, if someone wants to visit our institute, it's the Helmholtz Institute, or short HIO. Maybe it's a bit easier. And if someone wants to see what I do on LinkedIn, there's also, you can do that. Okay, thank you very much.
Thank you. Thank you, Olivia. Now we'll receive any questions. Any question, comment?
Thank you, Olivia, for the nice talk. I just have one question. It's about more of a clarification, and another question related to the sampling site. So, the first one, in the diversity data you show somewhere with the stack bar plot, you have swaps. I was just wondering, maybe you mentioned it, but I missed it. So the swaps are from different plant species. Is that correct? Is it from a swap from different plant species and or different sampling site, but within the same forest? And another question I have is related to sampling site. So what is interesting about Ivory Coast and Uganda? Why not somewhere in the Central Africa also, something related to that? Why particularly the West and completely the East? Thank you.
Okay. So for the leaf swaps, we also published the protocol, also for the flies, if someone wants to see it. And the idea is so we use leaves because that's the forest surface, right? So in a house, you would maybe swap the walls, but we say leaf swaps, but it's surface swaps. So we try in this 3 minutes that we are swabbing to cover as many leaves as possible. I hope that, and this includes different species, but we are not, we don't do any research on what kind of species. Although, of course, any species that, how do you say, makes it easier for animals to shed their DNA, so that have spikes or a bit rough, probably they will also have DNA, but we didn't investigate this in particular, but that could be interesting also in the future. And the second one, I already forgot. Why Ivory Coast and why Uganda? So for this, I have to say originally are like a disease primatology institute, and we have 2 field sites, one in Thai National Park that is known for the chimpanzee populations, and one in Kibale National Park that is also like a biodiverse hotspot for so many different monkey and ape species. And I think that's the reason why we started this work, also to see if especially those monkey and ape species are coming back once you restore the forest.
Any other question?
Yeah, thanks, Olivia, for the interesting talk. I wonder, regarding your different stages of the development, I didn't get it from your map. Do you have replicates of the different stages, and if so, how many?
Okay. So yeah, I didn't show that in the map. So the replicates will be each month, right? But for each month, the collection of each month varies from the material. So for the mosquitoes, we would do triplicates for 3 nights. So that would be 9 trapping events. For the leaf swabs, we actually found out that we can put 2 swabs in 1 tube. And we do that along a 4-kilometer transect. So we do that 5 times. So in total, we have 10 replicates. And for the flies, we're also doing 3 replicates. Yeah. So you mean like 2 times 100? Yeah, okay. No, we didn't do that. Also, the sites is not something I picked, but it builds upon work of Colin Chapman, ecologist and primatologist, that has been doing census on this spots for 30 years. So of course it can be different, and also I think, I don't know if you recognized on the mosquito abundant chart that the 2 ones that are 100 years old, they already look a little bit different. But I think ecological studies are always like it's like a fraction of how the forest looks like. So that's just how it is.
Thank you. Any other?
Thank you so much, Olivia. I have 2 concerns. One, it seems this component of eDNA is an innovation that has come up. I'll request you to say something small about it in general. Then 2, I would like to know how the restoration of biodiversity affects or links with the reduction of infectious what? You called them infectious what? Somewhere there I saw. Was it infectious diseases or something related to that?
Sorry, disease dynamics?
Yes, disease dynamics. I just need to know that connection or that linkage. Thank you.
Okay. So for eDNA, I think aquatic eDNA is, I mean, all of eDNA is very young, but of the eDNA, aquatic eDNA is the best studied, where I think terrestrial eDNA is still lacking, and you have different methods. The leaf swabs and the flies is only what we discovered works best for our questions. You could also use air filtering. There has been studies that hang air filters. You can do that active or passive to filter the DNA, but the idea is to collect the DNA that animals leave behind, or any organism. I hope that answers your question. And the second one I didn't really understand. Maybe you can. So the reduction of transmission risk, right? And you just have 2 species. You have 2 species of mosquitoes. So it's much more likely that this one affected Aedes mosquito would bite a human. But if you have the same species with 100 other species that are not capable to infect humans, the pathogen would therefore be, quote unquote, diluted. And that's what we're trying to prove. So we measure biodiversity because we believe with higher biodiversity, pathogen risk will be lowered. I don't know if that didn't answer your question. Maybe you have to. Okay, let's do it like this.
Yeah, thanks very much for your talk. I think this is very interesting because when I was listening to you, I remember of something that is happening in a village that I was born in, around 26 kilometers from here. So when we were young, we could not see monkeys around our farms, but now there are small monkeys that are really a menace in the village. So this actually made me think maybe there is something we should be doing because this is not normal. It has not happened since I was born. Those who are all older than me can attest of that. So we didn't have monkeys like in the villages coming. It was only in the forest. So since we have these monkeys coming even up to the homestead and stealing food and things like that, should we be doing such studies just to be sure that we are not waiting until things go wrong? And if so, what do you think should be done? Like, what is it that can be done 26 kilometers from here and areas? I'm sure it's not only my village. You've seen this happen in other villages. Small monkeys coming, and people are really concentrating on the fact that they destroy crops and maybe steal a few things. But we are not really thinking of what surveillance we could be doing in terms of disease and so on. So what do you think should be done in such a case?
Okay, thank you very much. Yeah, animal-human conflicts are a big thing, also in national parks. For this particular event, I don't know what happened. So I think that would be the first step also to find out why it changed. And if it's really a restoration method, if it's really because the bordering national park declines because people, I don't know, use timber or for other reasons. And these monkeys are really monkeys from the national park because that is another thing. If maybe they're coming from somewhere else, then also planting more trees maybe would not help. But to answer your question of the disease surveillance also, I think in general, whether it has to do with restoration or not, it is always good to integrate rapid disease surveillance, especially with monkeys and apes, because there's a lot of diseases we can share. And if they come and be close in the villages and humans have more contact with them, then I would say yes. If restoration is the answer, I cannot answer that at the moment.
Thank you. There might be other questions. If you have, feel free to still get to Olivia. Thank you very much and all the best in your PhD studies. Thank you. So we move to the next presenter, and Anne Laigong will be presenting on genomic characterization of human adenoviruses in patients presenting with febrile and respiratory infections at select hospitals in Kenya. So Anne is with us. Welcome.
With causing keratoconjunctivitis. So when we come down to the nomenclature, which is actually one of the largest part of this work, the adenoviruses have been shown to recombine quite often, and the recombination is what drives the evolution. So recombination of the capsid proteins, as I'd shown before, have been shown to bring up new genotypes. And the genotypes, the recombinant genotypes, have also been shown to be more, to cause severe illnesses. So here we have an example of adenovirus B, where if adenovirus species B, genotype 7, the penton of that virus recombines with the hexon of adenovirus B7 and the fiber of adenovirus B3, it brings up a new genotype, adenovirus B66. Same to B114 is a recombinant of adenovirus B7 penton. Hexon comes from adenovirus B3, and the fiber comes from adenovirus F3. So this is the current nomenclature of the adenovirus genotypes. This has been, so we have a consortium, it's called the Human Adenovirus Working Group. They're the ones who determine the naming, and they're also the ones who determine if a new genotype has occurred. So this is how the naming comes up. And as a result of the recombination, we have evolution of the adenoviruses. So where is the knowledge gap? So currently, genomic data that is critical for accurate genotyping is missing from Kenya and the larger East Africa. And for most studies, they use single gene typing. So they use either the penton region, the hexon or the fiber, mainly the hexon because it's the largest capsid protein in the virus. And as a result, you might be able to miss recombinant genotypes. So, and as I've shown, recombination is what drives the emergence of new genotypes. So the study objectives for our work was first to determine the diversity of adenovirus species and genotypes in patients with febrile and respiratory illness in select hospitals in Kenya. Then we also wanted to compare capsid-based gene typing, which is the current way of typing adenoviruses, with whole genome phylogenetic analysis. Then we also wanted to assess evidence of recombination events among the circulating HADV genotypes. So this work, we used samples collected between August 2024 to January 2026. So we had 8 sentinel surveillance hospitals. So we had in the different counties, so we have Trans Nzoia, Baringo, 2 in Kisumu, 1 in Migori, 2 in Lamu, and 1 in Mombasa. For this study, we got ethical approval from CERU and from Walter Reed Ethical Approval Board. So for the methodology, for sample processing, samples, nasopharyngeal swabs were collected from patients presenting with fever of above 38 degrees and 1 year of age, above 1 year of age. The nasopharyngeal swabs were taken to the lab in Kisumu, where we did automated extraction of total DNA using Kingfisher platform. Then we did a sequence library preparation using the Colibri kit, then enriched the libraries using the backup set probes. So these are probe-based capture design these are probe-based method that was designed at the Center for Infection in the Immunity at Columbia University. And these are probes that target approximately 2,000 non-vertebrate viruses, which now enables the detection and full genome recovery of viral diverse pathogens. For sequencing and analysis, we did sequencing, single-end sequencing on Illumina NextSeq 2000. Then for bioinformatic analysis, we did quality check and trimming. Then we opted for reference-guided assembly. Then we generated the consensus genome. Then for genotyping, as I said before, we were trying to look at the whole genome and the capsid, so that is what we did. Then for recombination, we tested using the RDP5 recombination detection program, using the 7 independent algorithms. The results were analyzed and visualized in Simplot 2. So looking at our results, in total we had 1,969 total nasal samples, of which 163 had adenovirus reads. From the 163, we were able to confidently speciate 58. Of the 58, we had 23 which were species B and 35 which was species C. So as you can see, for species B, of the 23, we were only able to resolve 22. So we were able to genotype 22. But for species C, we were only able to genotype 7. The 28 were unresolved. So when you look at the genotype samples demographic, of the 29, genotyped samples, the median age was 3 years, of which I'd said before, adenovirus mainly causes respiratory infections in children. There was not much difference when it came to gender. So we had 14 male samples, 15 female samples. And you can see the county distribution and the age distribution. The highest was Mombasa and Migori, and only one was positive in Trans-Nzoia. So looking at capsid-based typing, we did phylogenetic analysis. So the ones in red, I hope it's visible. The ones in red are the study samples. So I've highlighted where they cluster with. So we have those from the penton, the hexon, and the fiber. So we have our samples on the penton for adenovirus B, clustered with adenovirus B7. For C, we had a split between C5 and C89. So on the upper part here, we have adenovirus B, and then on the lower part, we had adenovirus C. So you can see also on the hexon, we had a division when it came to adenovirus B, where some clustered with B7 and B3. But for the adenovirus C, they all clustered with C89. Then for the fiber, we had all of them clustered with F3, and all of them also clustered with C89. So bringing in the capsid typing, I picked just 2 random samples that you can follow. So it's not visible, so I'll just call them sample 1 and sample 2. So we can see sample 1 on the penton, it clustered with adenovirus B7. On the hexon, at sample 1 now clusters with adenovirus B3. But on the hexon, it remains as B3. Sample 2 the same. On the penton, it was B7. On the hexon, it moved from it's retained B7, but on the fiber shifted to B3. So what does this tell us? So for sample 1, according to the current nomenclature, it would be classified as adenovirus B66 given the pattern of the capsid proteins. And for sample 2, it would be clustered as adenovirus B114, again given the pattern of the capsid proteins. So let's look at whole genome. This is a whole genome now that we got. For the same samples, you can see there's for adenovirus C, they all are C89. But for adenovirus B, we now have a split. I just illuminated on that part. We have those that's clustered with B114, B3, B66, and B7. So as we'd seen on the capsid typing, there was none that was B3, there was none that was B7, but from the whole genome now it introduces B3 and B7. So let's look at the two samples we had before, sample 1 and sample 2. Sample 1 now clusters with B3, and sample 2 now clusters with B7. So you see there's a discordance in the naming of the genotypes. So just that as summary, you can see from capsid typing, it was a B66, from whole genome B3, sample 2, capsid typing B114, whole genome B7. So this is just a breakdown of the results. So you can see from the capsid typing, 12 of them all clustered with B66, but from whole genome there was a split between 9 of them retained as B66, while 3 shifted to B7. Same to B114, 10 of them, all of them were clustered as B114, but on whole genome, there was a split again between B3 and B114. When it comes to adenovirus C, of the 35 that were detected, 7 were genotyped confidently, and of these, 5 showed concordance between capsid typing and whole genome, but for 2 of the samples, there was a discordance where they clustered with C5 on the penton gene, but C89 on the hexon fiber in the whole genome. So now this brings effect into genotyping, given that different genotypes show varying disease manifestations. So we need to be able to confidently say this is one genotype and this is the other genotype. But for now, we have a bit of discordance. So for recombination, we were able to detect one recombination event in our samples, which was a recombinant between adenovirus B7 and B114. And the breakpoint was located in the hexon region. So here we can see the one in pink, I don't know if it's visible, there's pink and brown. So the one in pink is B7. So you can see B7, it has 100% similarity at the beginning with B7, then it drops down, and B114 takes the rest. So all these were supported by the 7 algorithms in RDP5, and they had a p-value of less than 0.05. So in conclusion, adenovirus B and C still showed dominance in respiratory infections. And we have seen these limitations in capsid typing and whole genome typing, whereas from my standpoint, I believe that we should look at the whole genome to genotype the adenoviruses. Then we've also shown that there's active recombination, and as such, there's need for continued genomic surveillance. So what are the next steps with the project? So as we'd seen before, we lost a number of samples that were not able to be resolved. So the next step was to do targeted sequencing, targeted amplicon sequencing. We designed primers that target the whole genome and do long-read sequencing on ONT with the hope of improving genotype resolution and being able to classify more of the circulating adenovirus strains. I'd like to thank the following bodies, and thank you so much for listening.
Thank you. Thank you very much. Now I welcome any question or comment. Maybe I start off. We've known adenovirus giving us the platform to get the adenovectors and all that. So what could your findings be, or these are not the strains that we usually use for generation of the vectors?
So for vector generation, mostly we have adenovirus 5 that has been used a lot. That is one that has been studied. So from our results, we were not able to, apart from one sample that clustered with C5 on the penton, from whole genome, we did not have any C5. We only had the recombinant C89.
Yeah. Any question? Comment?
Thank you very much, Laigong. Now I remember where we met virtually. She's one of the potential bioinformaticians, and I'm really proud of what she just presented. My question is, I could see the global variants and then the ones in red. So those ones are not global. Are they unique to?
The ones in red were the study samples.
Okay.
Yeah, so the ones in black font were global, that used to give context to the tree.
Okay. Yeah. So what does this mean in terms of the treatment and vaccines? What does it mean? What does it tell us?
So as Adad mentioned, we have different species. The different genotypes show different disease manifestation. And we have certain genotypes that have been shown to cause outbreaks, especially in closed spaces. So currently, we only have one vaccine for adenovirus, and it targets adenovirus E4. So most treatment right now is mostly just normal antiviral treatment. Antiviral treatment. So it's just like they treat like the cold for adenoviruses. But for those that cause gastroenteritis, there is also a potential for outbreak. There was an outbreak in 2024 in Kilifi, which had it was for gastroenteritis, and then we also had one in February in Mombasa, which was respiratory, and the causative agent was adenovirus. But now, in a case like that, since we did not get the samples, they did not really genotype. We can't tell what's there. So this is mainly for surveillance and being ahead of the curve. So if we see maybe one adenovirus B7, and that has been shown to cause outbreak, we can be able to get ahead of that. Yeah. Have I answered the question?
Yeah, you've really answered well. Thank you very much, and keep it up. Yeah.
Thank you so much.
Thank you. Let's applaud Anne. Thank you very much. So we are going on well. We are now proceeding to the last presenter, under the sub-theme genomics in diagnosis, surveillance, and management of disease and epidemics. So Catherine Nasambu will be the next presenter. And our talk is titled A Metagenomics Assessment of Wolbachia Bacteria as Potential Biocontrol of Anopheles gambiae and Anopheles arabiensis malaria vectors in Siaya County, Kenya. Catherine, are you with us? Welcome.
Thank you, Dr. Good afternoon, everyone. I'd like to appreciate for the organization of this symposium and for the opportunity for me to present this project. My name is Catherine Silali Nasambu, and I'm a fourth-year student at the University of Eldoret. And my topic of study is the molecular assessment of Wolbachia bacteria as a potential biological control of My topic of study is about molecular assessment of Wolbachia bacteria as a potential biological control of Anopheles gambiae sensulato in Siaya County, Kenya. I was privileged to do this project at the KEMRI, Kisumu Entomology Lab, where I got the idea from as an attachee. I got the idea from the KEMRI, whereby I attended a conference whereby someone was presenting on Wolbachia bacteria in Anopheles demilioni, which is a highland mosquito. And studies had shown that most of the Anopheles mosquito don't have Wolbachia. And for the first time, he presented Wolbachia bacteria in the Anopheles species, and it came up as a knowledge gap in the primary malaria vectors, such as the Anopheles gambiae sensulato. So according to the World Health Organization report of 2025, malaria remains one of the most significant public burden, especially in sub-Saharan Africa, where the environmental and socioeconomic factors favor the transmission of the malaria vectors such as Anopheles gambiae sensulato. So Anopheles gambiae sensulato is a complex of species where these species are morphologically similar but different genetically. And various control strategies have been taken into place, but they have been threatened by the increase in insecticide, which has led to the need for alternative approaches. And Wolbachia bacteria is such an alternative because of its ability of vertical transmission where it can rapidly spread in populations, and it can also inhibit pathogen development in the host, and its mechanism in cytoplasmic incompatibility described in this figure, where when a male infected mosquito mates with a female, uninfected or infected with another Wolbachia species, we have an early embryonic death. So this study basically investigated the occurrence of Wolbachia in the Anopheles gambiae sensulato species, and it also investigated the association between the Wolbachia bacteria and the prevalence of Plasmodium parasites. As I said earlier, one of the major problems faced by the control strategies is insecticide resistance, which has reduced the effectiveness of the indoor residual spraying, and I'm sorry. The spread of insecticide resistance is in the malaria vector, primary malaria vectors, has led to reduced effectiveness of the current strategies. And Wolbachia offers such an alternative. This bacteria has been successfully used in Aedes aegypti mosquito, whereby it has been used to block viral pathogens such as dengue, chikungunya, and Zika. So the objective of this study was to evaluate the prevalence and interaction of Wolbachia and Plasmodium infection in Anopheles gambiae SL populations from Siaya County. The specific objectives were to molecularly identify the Anopheles mosquitoes collected from the field, and to determine and confirm the presence of Wolbachia in the collected samples, and then to estimate the prevalence of the Plasmodium falciparum infections. The hypothesis of the study, the null hypothesis was stated that there is no significant association between Wolbachia infection and Plasmodium prevalence in Anopheles gambiae sensulato populations in Siaya County, Kenya. And the alternative hypothesis was Wolbachia uninfected Anopheles gambiae have significantly higher Plasmodium infection rates compared to Wolbachia infected mosquitoes. And the study area was Siaya County in 3 sub-counties, the Alego, Usoga, and Rarienda. I didn't participate in the field collection. I acquired the samples from the entomology lab, where we did morphological identification of the mosquitoes to classify them into gambiae sensulato, using the dichotomous key developed by Gallis and Coetze and a stereo microscope. The identified samples were then confirmed molecularly by screening the ITS2 region to differentiate the 2 sibling species in Gambiae sensulato, that is the Anopheles gambiae sensostricto and Anopheles arabiensis. After the identification of the 2 species, they were screened for Wolbachia, where 2 molecular approaches were taken. First, the 16S RNA the first approach was the use of 16 RNA by use of endpoint and real-time PCR, and then the samples that confirmed positive were further confirmed by screening the WSP gene by the conventional PCR. Then all the samples, both the Wolbachia positive and the Wolbachia negative, were screened for Plasmodium prevalence. And after which, a correlation between the Wolbachia positive, Wolbachia status and Plasmodium prevalence was done using the Fisher's exact test to assess the association between the two. And my results were based on the three specific objectives, of which my first objective was to molecularly identify the two species, Anopheles gambiensis sensu stricto and Anopheles arabiensis. And as the gel image shows, the first two bands are the positive control for Anopheles gambiensis sensu stricto and Anopheles arabiensis respectively. And in the middle we have the DNA ladder. And we did pick 95 of each intentionally. It is important to note that these species are not evenly distributed in Siaya County. This was an intentional distribution for easy analysis. And we came up with 95 of Anopheles gambiensis sensu stricto and Anopheles gambiensis arabiensis. So after identifying the two sibling species, we went ahead to screen for Wolbachia. And as you see in the image, the first two curves are positive control for Wolbachia, whereby the samples that tested positive for Wolbachia showed a clear amplification curve as shown. And they were further confirmed by running a conventional PCR, and the PCR products were resolved on gel electrophoresis. Though I didn't show the results for the bands, but the table shows the outcome of the Wolbachia status for the two sibling species, whereby we had Anopheles arabiensis, 13 of the 95 testing positive for Wolbachia, and 45 of the Anopheles gambiensis SS testing positive for Wolbachia. As you can see, Anopheles gambiensis sensu stricto has a higher prevalence for Wolbachia as compared to Anopheles arabiensis. These results suggested that Wolbachia distribution was not uniform in these two species, and it reflected that the species-specific differences, which could be due to host compatibility or bacterial density or transmission in the two sibling species. And after the detection of Wolbachia, all the 95 arabiensis and 95 gambi were screened for Plasmodium prevalence. And as you can see in this figure, we had a few detection for Plasmodium, whereby none of the Wolbachia positive arabiensis tested positive for Plasmodium. And for the Anopheles gambiensis sensu stricto, we had 3 Wolbachia positive testing positive for Plasmodium and 2 Wolbachia negative testing positive for Plasmodium. when we go back to Arabiensis, the absence of Plasmodium, the absence in Plasmodium in Wolbachia positive Arabiensis was suggestive of the possible protective role of Wolbachia. However, as we can see, the Plasmodium prevalence in these two species is very low. And I felt that the results were not that conclusive, but suggestive. And when we did a combination of the two, we had only only 3 of the Wolbachia positive testing, positive for Plasmodium, and only 6 testing positive for Wolbachia negative. Sorry. So the higher detection of Wolbachia rates in Anopheles gambi as compared to Anopheles arabiensis in this study implied species differences in Wolbachia infection, and due to the lower bacterial densities, differences in the mosquitoes' behavior and host symbiote compatibility, as well as transmission efficiency, implied that there is higher potential for the Wolbachia to be spread in Anopheles arabiensis as compared to Anopheles gambi as compared to Anopheles arabiensis. My study is similar to other studies where they screened for Wolbachia and they got low titers of Wolbachia, and many studies have reported inconsistencies in Wolbachia infection in the Anopheles gambi sensulato. So based on my study, we concluded that Anopheles arabiensis and Anopheles gambi are present in Siaya County, and natural Wolbachia infections were detected in this species, however, in low densities, and there was no significant association between the observed Wolbachia infection and Plasmodium prevalence. And based on the study, the following recommendations were made. The increase in sample sizes, particularly the Plasmodium positive mosquitoes, to improve statistical power. Then to perform Wolbachia strain typing, genotyping to identify circulating strains in the study. Because there is a wide range of Wolbachia strains. Some are stable, while others are unstable. So when you do genotyping and characterization of Wolbachia, you'll get to learn of the stable strains which can be used in the Plasmodium suppression in the mosquitoes. In addition to that, considering species-specific differences, as we've seen, Wolbachia prevalence was higher in Anopheles gambi sensu stricto as compared to Anopheles arabiensis. So future studies should consider species specificity in malaria vector control, and they can also explore transinfection experiments, because most of the studies that were successful with Wolbachia, they did transinfection experiments using stable strains from other arthropods such as the Drosophila melanongaster. And finally, I would love to acknowledge Dr. Chemoyua, Professor Moses Ngewa, and Mr. Cyprian for their support in this project. Thank you.
Thank you, Catherine. Thank you. Now we'll welcome the floor to some questions. Any question, comments? Yes.
Thank you for the very interesting study. And I mean, your data pretty clear-cut showed there is no association between the naturally occurring Wolbachia and Plasmodium. That said, I mean, you mentioned at the very end that this hope to mitigate Plasmodium by Wolbachia may also relate to heterospecific Wolbachia strains, right? So that would be perhaps another story if the Anopheles would be infected with Wolbachia, which they are not used to, or am I mistaking that?
I'm sorry, I didn't get the question right.
Shall I repeat the essence?
Yeah, kindly.
Yeah, I mean, the essence is, as I understood your sampling design, you were looking for association between naturally occurring Wolbachia and naturally occurring Plasmodium, and there was no association. That's
there wasn't any
right.
association the two.
And but it's hope to be able to mitigate Plasmodium by Wolbachia. They may use other Wolbachia strains, right?
Yes.
And then it could be a different story, in fact.
Okay, we have various strains of Wolbachia, and some are stable enough to block the pathogens in the hosts, but others are not stable enough. So for my study, I didn't characterize the Wolbachia to see which strain is stable and which one is unstable. Yeah. And as the study in Aedes aegypti, they used strains from the Drosophila and they transinfected it into the Aedes mosquitoes, and it was really successful in blocking the viral pathogens. Yes.
Any other question?
Thank you very much, Catherine. Mine is not a question. I simply need to appreciate
Thank you.
the presentation and the courage. I understand you are an undergraduate student. Surely the courage to get here together. Thank you so much.
Thank you.
Maybe one more? If not, let me close with one question to you. From your literature, maybe this is, of course, you're still going on with your studies. How could the bacteria be actually affecting the Plasmodium? You said it interferes with its development. I don't know if you have found anything. How is it actually maybe affecting the Plasmodium?
Okay, in my introduction, I said it's Wolbachia ability to prevent pathogen development. It can do this through competition of resources. So it deprives the pathogen in the host with nutrients, and the pathogen will eventually die. Yes.
Okay. Thank you. Let's applaud Catherine once more. Thank you very much. So at this point, I'll say we are done with the presentation under the last session, which is session 6, which was on genomics in diagnosis, surveillance, and management of diseases and epidemics. And again, I'll say thank you to all the keynote speakers under this session, Professor Anisep, Dr. Torore, and all the participants, all the presenters. Thank you very much. It was my pleasure again, and thank you very much. Now, I'll take this opportunity to welcome the next moderators, Dr. Chemoywa, Dr. Baraza, to guide us on the way forward and part of the closing remarks. Thank you.
Good evening. I want to take this opportunity to welcome our DVC Planning and Research, Professor Aburu, and acknowledge the presence of our keynote speakers and all the delegates here. We are so happy to have had a good presentation over the 4 days. And I know presentations are over, and part of way forward, which is not really way forward, we chose to maybe look at, discuss with you, or rather show you our lab, because we have not had a chance to go to the lab. So allow me to request for the slides in the email, the last slides from Emily. Yes, Asimwa. Yes, so I know some of us have been to the University of Eldoret, others have not been. We'll arrange for those who want to go and visit the lab and visit the university where, when we have a break now, between now and the evening event. We will do that for a few who had requested. Those who are still around, you'll have time to go to the university even tomorrow and see where it is. But we have a main campus, which is in Eldoret Ziwa Road, a few kilometers from here, about 10 to 20 minutes drive. And we also have a town campus here and in Nairobi. So what I want to present to you is our genomic lab, which is managed by Dr. Barasa and team, with our DVC being involved fully and under research office. And we've had a chance to launch it in a few years ago, and we've had a few projects going on. I don't know whether the presentation slides are there. Not yet. So meanwhile, as we do that, We've had a chance to interact a lot with a few of our keynote speakers, and in the genomic lab, we've had, like we said earlier, Prof. Ralph in the lab training our postgraduate students, both in hands-on lab activities and also bioinformatics. We've had a few of those trainings, and it has really, that's the reason why we have our postgraduate students having hands-on research in the lab. We've learned a lot. There's still more because we have many students who have interest in employing the molecular tools in their studies, both diversity studies and also crop science, animal science, fisheries. We have many of those. So we, at the Genomic Lab, we host not only the fisheries, but also students from various disciplines that cut across. For example, I'm from biological sciences, and so we have students from biological sciences accessing the lab. We also have students in crop science accessing the lab as well, and students from agriculture, students from fisheries. Those are the kind of, and chemistry. I know Sharon is looking at me and she's saying, yeah, I was in the lab. So we were with Sharon in the lab, and many more. So we wanted to let you know that we have capacity to host students, postgraduate students, especially in our labs. My slides are a bit large because it has the pictures of the university, and you understand. Yes. So part of what is in the presentation I've already mentioned, so it will not take too long for it. So those who will not have time, that is our university, that's the main campus. And yeah. So we said earlier it was launched, and it's funded by the National Research Fund, Kenya National Research Fund. And those managing the lab is actually Dr. Baraza. Professor Aburu is with us fully on this research. And we have objectives of our research lab: fish genetics and genomic research and training lab. We're actually focusing on promoting the university strategic plan and objectives, and some of them cut across innovation, research, training, capacity building. So we want to enhance quality teaching, research, and innovation as one of our key objectives. We want to position the university in such a way that we are the best research hub within the region and within the continent, such to an international level, because we have a state-of-art lab. Some of the equipment that we have in the lab, for those who will have interest to bring students to come for attachments and research, we have all molecular equipment that can manage all the way from. We also have, apart from the molecular lab equipment, growth chamber in our lab. We also have modern dissecting microscope and compound microscope, which are part of some of the research grants that other staff have acquired over time, because our lab positions us well that if we want to do molecular work and we need more funds, we are able to bid our request for grants because we have a lab. So if we really need molecular reagents or consumables, then it positions us better. We have some of our colleagues who have had grants within us and have come to the lab to do the work. So of course, I'll just take you through pictures of all that equipment in the lab. That's our PCR bay. We have the growth chamber section. We have the deep freezer sections. So when we have samples of students or researchers who need negative 80 to 86, we have we have that capacity, as well as all that. What we are lacking, we will look at as part of our challenges. Apart from the equipment we've been able to host, some of our researchers have been able to host renowned researchers. One of the group of collaborators that we've had is researchers that came along in the project of Dr. Selina Rono and myself, and Dr. Barasa was in, and other collaborators, where we had a partnership with the University of Royal Holloway and University of West London, and on the project that Selina and Sylvia presented earlier. We also have had other renowned researchers and collaborators we have is the team of Professor Ralph and his research team who were in the lab, and they partner with Dr. Barasa in the projects we have, and we've had capacity building on the molecular techniques and bioinformatics. So we've had trainings for students in the lab. This is our lab. Basically, this is our working area in the lab, where students, when they bring samples, we place them there and we process them before taking them for PCR and gel electrophoresis. We've had trainings on hands-on. This is presented earlier during this workshop, and with Professor Ralph. This is also a continuation of the same. We had bioinformatics training here at the main campus, the town campus, where we were taught how to handle any molecular data, all the way from quantifying them, not really quantifying, looking at them, screening them, and trimming, all the way to the bioinformatics. So those are some of the few trainings and workshops we've had. I told you earlier, we host postgraduate students, and over the years we've had students graduating. That's hosting DOSI, looking at scarabs, the insects, This is Hilda, the barcoding of Enteromia species in Lake Victoria. This is Ezra, who did his work on genetic diversity and also testing the primers within the lab. Other researchers and students who have been in our lab include Naomi. She was here yesterday for those who interacted with her. Sharon is somewhere there. And this is Sylvia, always in microbiology, making sure that she's safe when she's pipetting, all that. All those have at least interacted with the genomic lab, including Edith. Edith presented earlier on her work in fecal oral zoonotic pathogen. If you remember, this Daisy, looking at Orichromys esculentus, earlier she presented her work, and she's still going on. These are students who are going on in the lab, still processing the data, still finalizing. We also have Purity, who presented earlier on species identification of Orichromys niloticus earlier, and she presented very well, including one student who is new in the lab, currently in the lab using the growth chamber. We have, for crop science, we have the crop chamber, and that's Timothy Kiagu, who is evaluating temperature, humidity interactions affecting affecting germination and disease severity in wheat stem rust. So he's still going on, doing some of the work some of the work is done within the greenhouses, and they process their work, and then they come to the lab where they can access the growth chambers. So basically, those are few of our students. We still have capacity to host more. And so our potential contribution of the lab, of the Fish Genomics and Genetic Lab, is to serve as an exclusive research hub in the region. We also want to provide opportunity to collaborators and collaborative multidisciplinary research is what we really are focusing on now, of course, and also, yeah, we want to offer resources that we want to have the Genomic Lab a resource that can offer consultancy and investigative services to the community. The brain behind the Fish Genetics and Genomic Lab was to offer, resolve challenges that are within the community, and that's what we want to focus on as a lab. As much as what we have done so far, so much of it is been postgraduate training and research, but we want to focus on CSR so that our lab is able to solve the community challenges that we have within the community. That's what the team of Dr. Baraza and others are looking at, even resolving challenges to do with fingerlings, distribution within Uasin Gishu County, and ensuring that the farmers get the correct seed. We also have opportunity for internship. Of course, it's limited to the postgraduate students who are specialized in molecular techniques. Yes. So challenges that we face, not so many, but some of the challenges that all of us face in our labs, including frequent power failures. At times a student is working, and by the time we get the power back, it is a challenge. Delay supply of reagents, of course, stringent procedures when we are looking for funds, costly reagents, limited local funding. At times when we need to calibrate equipment, it is a challenge. And so we are always told that we have to work hard to write proposals that can bring grants to service the lab. So that's the challenge that the research office has given us as researchers to ensure that we have funding that can sustain the lab. So in conclusion, we want to foster partnership. That's why in part of this workshop here, we have you as our collaborators and our potential collaborators in the lab, because we want to further equip the lab infrastructure and also equip cutting-edge instruments and also train our researchers to be skilled, to be the best, so that we can offer our best. Of course, train our students. We've seen earlier possibility of capacity building, including Africa Genome Biome, and others. So if you have any link that we can step into, we are free to pull resources together and ensure that our expertise can unlock full potential of genomic in health, agriculture, and environmental conservation. We definitely want to do that as a lab, as a unit in the university. We recommend collaboration among institutions and scientists in the region, and also more funding by both local and international funding institutions. Not just region, we also want to expand, of course, and get more funding to support the research in our lab. So this lab was funded by NRF, and we want to really thank the NRF and University of Eldoret for giving us an opportunity to be in that lab and to manage it. Thank you so much. I will stop there and invite Dr. Barasa. Maybe a few things I left out, but Dr. Barasa will be able to discuss further. And after that, he will welcome way forward discussion.
Yeah, so thank you, Emily, for that elaborate expose of our lab. So that is where we are, what we have. That was just an overview, and I believe there is still a lot to build on and enhance what we have and the collaborations. So the way forward, the next section, we just want to look briefly at the way forward. And to begin it off, I just wish to bring to your attention or to your notice that some of us have been inquiring whether we will have a chance to publish some of our papers presented at this symposium. So we've made an inquiry with the society, SMBE, and we've been informed that tentatively we can begin to prepare our manuscripts. They are exploring options that can accommodate our papers. Yeah, so those who have an interest to do so, to publish in SMBE-hosted journals, please just begin preparing your manuscripts as we wait for more details. They have tentatively suggested open submissions because they have not been having special issues for their journals. I think, Ralph, you are aware of this. But they have also said that there is a possibility of considering a special issue to accommodate our symposium. Yeah, so these two options are still under discussion, and we will get a clearer way in the coming weeks. So meanwhile, you just need to begin working on your papers. But whichever option we are given, all the papers submitted will go through peer review. And as you might be well aware, their review is very, very stringent. Yeah, it's rather stringent, but I believe we can make. Yeah, we can make. So that is part of the way forward, and I just bring it to your notice that we can begin to work on this and to see how we can now increase our own visibility as researchers, as early career scientists, especially for students, and the rest. So on that note, maybe I may just open the floor briefly for comments on potential for collaboration, especially amongst this group, having looked at the presentations, the diverse fields, the data sets that we've seen, and the potential for most of the presenters. So what would be the way forward? What would be the way forward in terms of collaboration, network building, potential for training for PhD students, even master students, and such opportunities. So on this note, I might just welcome Professor Ralph to make very brief comments on what he views as a potential for collaboration, or what opportunities does he foresee from this symposium and chance. Thank you.
Thank you very much. This is, of course, a personal subjective statement, but I think the first I can start, and I hope that is not only me, but everybody here in the room. I think these were great 3 days, and we can thank the organizers a lot. They have done a tremendous job. It was interesting science, but it was also interesting sheds around the science. So, and I indeed think it's venues and meetings like these which are essential, because in two aspects they actually bridge across countries, but also across career stages. And it's also important to have these formats not too large, so that actually early career scientists can approach the senior scientists and speak on equal grounds, and thereby exploring possibilities. I think what we have seen the last days is a lot is going on in African genomics, which is very good. At the same time, the challenges are huge. I mean, in the morning we heard sequence 100,000 species. Then a few lectures later, we learned about personalized medicine, and there you may say sequence 100,000 African humans could also be a good idea. So where to start? There's so many things which can be done. And I think, I mean, you could argue it's a bit like breadth versus depth. Shall we go in depth with a certain species, or shall we try to tackle all? Ideally, we want to do all at the same time. but that may not be possible. So there is this breadth versus depth thing. And I think actually, think big is good, but start small, make it concrete, let things really happen. I mean, we have this small study on the Turkana tilapia, which attracted a lot of interest. You may say that does not save the world. That's only one of 100,000 species, but still, it is concrete. It's a concrete student involved, learning things, and larger things can develop from that. I think and it's perhaps a generic statement, and perhaps also comes without saying, but I think it's important that there is, among the PIs of the different countries, that there's mutual trust. There's also a clear benefit for all partners, and also sympathy helps. I mean, if you like your partner, that also helps more than if you don't like. And then the second step, which is essential, that we involve early career scientists, because the PIs typically, they're in the planning, they're perhaps in the fundraising, but they're typically not the ones who are really doing the work. It is done by early career scientists. So it's important, I think, to exchange people, to have local scientists learning elsewhere, but coming back to Africa and apply that here. Focus on local capacity building. It seems to me funding is often coming from outside. I mean, the lab here is a laudable exception that Kenyan Science Foundation really substantially invested, and I think it was a good investment. But many of the funds come from outside, and that may be very country specific. I mean, I see it from Germany. There are several funding schemes for various career stages, but other countries, UK, US, others may have that also. That can be exploited, and I think I would not trust a single line there. Just make contacts with whomever you trust or with whomever you can well collaborate. That's perhaps a very generic statement, so you have perhaps hoped for that I'm more specific what to actually do. But I think it is important to know each other, to invest in meetings like these, and also to be, as I said, make it concrete, make things really happen, not dream of too huge things, but start with concrete projects which really happen, where really scholars are involved. And then, meanwhile, you can think of larger projects, and every now and then you hear a call. I mean, coming from the European Union, every now there are European calls. If one is prepared already, knows the actual stakeholders, you may have an advantage in applying because you're quicker off. And that's perhaps what I can say. And for me at least, and that's again subjective, what is the mutual benefit of this? It's of course the science, but I also love to be in Kenya. So that's also really an additional benefit to have the ability to be here and be welcomed here. Thank you very much.
Thank you, Ralph, very much for those good comments, encouraging comments. Yeah, so may I also just welcome Professor Paul Gropler to make his views, very brief views on what he views as a potential for collaboration.
Thank you, James. And let me also start by saying that I've enjoyed the last 3 days tremendously. I think it's been a nice, close-knit group with a very large amount of exchange of ideas, and I think that was a very positive aspect. Yes, in terms of collaboration and the way forward, Prof. Ralph touched on something very, very important, that it's all very well to talk about large-scale collaborations, but I fully agree that aim for things that are small and very important, that has a fixed endpoint. Take a small project that you know is going to have an output in the form of a student or a publication in 18 months or 2 years, and we get concrete, everybody gets concrete benefit from it. But just some additional ideas. Yes, in terms of research, I think everybody sat listening to presentations, and in some presentation picked up something that says that speaks to what I am doing. And that is, of course, where collaboration starts. And collaboration can take many forms. at a very basic level, it could just be to start talking to people that's doing the same thing as yourself is doing in terms and exploring ways to analyze your data, maybe new ways to analyze it. A step ahead of that, or a step further than that, is possible access to research sites. You may sit here and look at somebody's work and realize that that site has potential for the kind of work that I'm doing. Maybe I should talk to that person and sample something at that site. And of course, access to material. Very often there's additional data that can be derived from already sampled material. So that is also a possibility. Yes, if perhaps I can throw in some specifics here, just to plant some ideas with you. There's been very few mammal presentations at this conference, but I do want to plant some ideas there, and you can perhaps talk to me. Or some things can develop, and there's a very good reason why such work can be useful, apart from the generation of data. If I look at the South African system, we're really sitting at a point where it is ridiculously difficult. Is this being recorded? But it's really difficult to import samples, not because of the traditional permitting things. That's fine. We go through that. But for veterinary reasons, there's extreme caution about bringing any kind of material, biological material, into the country. And I think a very good way to tackle that would be in-country analysis, which immediately brings new benefits. So hypothetically, for me to start a mammal project in Kenya, and then having the analysis done here and involving local students and staff in that analysis, followed by all the other steps of research, I think that will be extremely valuable for me and for others to move beyond moving samples back to my unit, but actually doing both the collection and the analysis in country. Yeah, so if I can maybe just plant a couple of ideas for you to think of. Although I presented a large spectrum of work that we do in my unit, I think one or two that stands out in terms of suitability for application in Central and East Africa. I think perhaps the one that for me really stands out is using DNA-based census techniques for elusive animals. And we're doing it on some antelope in South Africa. And if you think of forested areas here, I think there's vast potential for DNA-based census techniques. So I would be quite interested to talk about that. I was thinking of porcupines. Porcupines are not so elusive, but they are difficult in this part of the world because you sit in the middle. There's two species of porcupines. Right to the north you have crested porcupines, and in the very, very south of Kenya you have Cape porcupines. But still, if somebody feels like doing some work on porcupines, I would be very interested. Another one that links closely or more closely with the aquatic focus of your lab is freshwater mussels. I've done actually quite a lot of work on freshwater mussels also in the United States, and it's an incredibly neglected group from a genetic point of view. There's taxonomic descriptions of them, but genetic studies of freshwater mussels are few and far between. So linking that to some of your fish sites could actually be a nice addition to some projects. Yeah, and then moving on to fish itself, we could look at continuing some of our catfish work and finding new angles there. In terms of training, yes, there are various possibilities there. Hosting of students, and that working both ways, hosting of students this side or hosting students elsewhere, could be very valuable. And with that co-supervision of students, again going both way. Yes, in terms of training on site, I don't want to say too much about this because it's an area that is well established by Potsdam. So yes, that is running. But if there is an opportunity, then certainly I have one or two very good bioinformatics people in my unit. So one could think of becoming involved if there is room for that. One could take this kind of thing a lot further and formalize short courses if there is a need for that. At my university, there's very much a push for that. In fact, we're no longer allowed to present ad hoc short courses, but so things are formalized, but then it goes with an official small qualification as well. So that's maybe also something that could be explored. Thank you, James.
Thank you, Professor Ralph and Professor Paul Grobler. Let's applaud them once again. So as we move towards the end, this session is very important. Both professors run excellent labs for genomics, and it's always good if you can link up with them. Yeah, discuss your research ideas, discuss the opportunities that are available there, and see if your idea can be attractive to them, then open collaborations and see the way forward. I know that we've had long discussions. Professor Cyprian Katongo from Zambia comes from an area that is rich in fish biodiversity, and is always very willing to bring on collaborations. And those people in the fields of cichlids, Professor Katongo provides an important avenue of collaboration because of Lake Malawi, Lake Tanganyika, and the internal lakes in Zambia, and the rivers there. You saw what he said yesterday, so always feel free to reach him. Dr. Rispa is at the medical school here. I hadn't interacted with her before, but I've seen that she's running a very rich research program at the medical school, and we would be very happy if we can begin to do things together. So these are the opportunities. Please feel free to see how you can harness them for your individual benefit. But before we move to the next section, perhaps I may just give a chance if there is any clarification from the floor or any question from the floor or any additional comments. Yes, Prof.
Says Symposium under the International Union of Biochemistry and Molecular Biology. It will be biochemistry and molecular biology. It will be similar to this African. So we might extend one or two invitations around for people to consider. Thank you very much.
Thank you, Prof. Any other comments? Yes.
Thank you very much. Mine is a comment and a suggestion. It's really impressive to see University of Eldoret having to start small with some functional lab that was funded. And I love to say we were competing with the team, with Dr. Baraza. We were two teams from University of Eldoret. He won, but I was happy. So I'm happy to see that the genomics lab is there. Maybe I'll have one more university in some time too. So the suggestion is, with that functionality, maybe it's good if it doesn't just become internal, if you could open up and even let us know how we can bring our own students some funds so that we can make it, more sustainable and have things coming, projects coming through the platform that you have at University of Eldoret. Thank you.
Thank you, Vivian. valid comments, and we always welcome students to our lab. It's quite open. In fact, it's supposed to be a regional lab, so all scientists and all students are welcome, and we want to build on those collaborations. Thank you. Is there any other comment before we move to the next section? Yeah, so if there is no comment from the floor, maybe we just want to transition to the next section where we begin the closing ceremony. Yeah, so within our midst, our Deputy Vice Chancellor, in charge of Planning, Research and Extension, has come to see the closure of this symposium, and we are happy to welcome him. So Prof, feel welcome, Professor Aburu. So for the last 3 days, we've had very illuminating discussions and presentations. I think we've seen data sets from the molecular field, across fields, environment, health, fisheries, natural resource management, crops, animal breeding, and all fields. And we've had good presentations, actually excellent presentations from many of us, including students, and I believe that they have really learned a lot. Yeah, they have really learned a lot and even gained skills, in presentation and such important things. So we want to take this chance to welcome you to make a few remarks, then you close. But we were more than this. Some colleagues, some visitors, guests have left because of their schedules. Some delegates presented online, because of the inability to travel to Kenya. And some guests also presented online. So we are happy. We will be sending these recordings to all of you. But so we just want to bring this to a closure by welcoming the DVC to make his remarks and close the function. So let's welcome the DVC, Professor Aburu. So Prof, feel welcome.
Okay, good afternoon, ladies and gentlemen. Some have refused to greet me. Good afternoon, ladies and gentlemen. Okay, thank you so much, dear colleagues. I'm happy to be here, though I would have wanted to be with you from the beginning, but I was away until yesterday night, and today I thought I would be with you the whole day. But we've been having some Senate meetings, so I have missed quite a lot because I really hate appearing as a politician. It's politicians who appear to close what they don't know what has happened. So today, Baraza has put me in the same situation of closing what I don't know what has happened. But anyway, I understand because since the organization of this started, he came and told me when it was still proposed, and throughout the planning, I think we have been consulting. And we just want to thank you so much for dedicating your time to come so that we'll make this symposium a success. As a University of Eldoret, I'm sure you may have been informed for those who are visitors. We are a small institution, a young institution that came from Moi University. And we were a campus of Moi, and we are still growing. The one thing that Moi University gave us was we were the science campus. So when they went, we remained with everything. So we are more senior than them in science than other courses because they had given us all the facilities, and we thank Moi University as our mother, and we continue working together. They sometimes contribute some of the stuff to us,
And even some of us, like Tuwei, who is here, ran away just to go back, to be back with the mother. So, Daktari, I hope you are enjoying yourself with the mother. Okay. Secondly, please feel more welcome. We would have really have loved for you to also get some time to come and visit us. But we know that time is not very easy to get all that time. But we appreciate you, all of you coming and being here for this time. Before I read my closing remarks, I just want to also weigh in onto the last comments that were being made. As a university, we are very open to collaboration. And we have been trying to widen our collaborations so that we can have more and more. So much more of projects and research projects, both locally and internationally. And this lab, as Dr. Baraz had said, this lab is for the region. Anybody who would like to do his research or her research there is welcome. And once you come through Dr. Baraz and his team, you will be allowed to do your research here, because that's the purpose why the lab was created in this region. I want to applaud our international friends and guests and scientists who are here, and also our students. I think we have seen a lot that has been going on, and we have room to grow. And it's always good to grow when you have a mentor, and we thank the mentors who have been mentoring us in different ways through trainings and things like that, and we highly appreciate. So with those few remarks, just allow me to go to the most boring part of closing a ceremony. Are we ready? Yeah, okay. So distinguished scientists and researchers, representatives from the Society for Molecular Biology and Evolution, guests, staff, students, ladies and gentlemen, on behalf of the University of Eldoret Management, allow me to sincerely thank all participants, organizers, facilitators, and partners who have contributed to the success of this regional symposium on evolutionary genomics for Africa 2026. Over the past few days, we have witnessed rich scientific discussions, knowledge exchange, meaningful engagements among researchers, students, and experts drawn from different countries and institutions across the world. The symposium has demonstrated the growing importance of evolutionary genomics and molecular sciences in addressing emerging global and regional challenges, including antimicrobial resistance, biodiversity conservation, food security, public health, and climate change adaptation. We particularly appreciate the Society for Molecular Biology and Evolution for the support accorded to the University of Eldoret and for choosing our university as the venue for this important regional scientific meeting. The interactions and collaborations initiated during this symposium will undoubtedly strengthen research, networks, mentorship opportunities, postgraduate training, and future joint research initiatives among institutions within Africa and beyond. And as I said earlier, at the University of Eldoret, we are very much open to collaborations. Currently, as we speak, we have so many collaborations in different areas. But in this area that you are discussing here, there are not very many, and we will be much interested, at least to extend, so that any collaboration, like has been said with our colleagues, we will highly appreciate, so that we can be able to do things together. We are really interested at this stage in internationalization and doing things together with the whole world, so that we can strengthen our staff, our students, and also to be able to solve the myriad of problems that are there within this region. As a university, we remain committed to supporting high-impact research and innovation through our existing facilities and centers, including the Fish Genomics Research Laboratory and the Biotechnology Center, while continuously seeking strategic partnerships that advance scientific discovery and capacity building. I recognize what our colleague talked about in terms of training, capacity building. Apart from the research activities, we are very much open, either coming up with joint courses that can be offered online, or any other form of capacity building which is feasible and which is possible. We very much appreciate and will very much encourage. We wish to encourage all participants to sustain the connections established here, continue collaborative engagements, publish the outcomes of your research, and translate scientific findings into practical solutions that benefits communities and the society. To our international visitors, thank you for taking the time to join us in Eldoret, the land of champions. So the fact that you came here, we want to declare you a champion before you go back. Because when you arrive at the land of champions, you become a champion automatically. So please don't be surprised when you go back and you start running faster than you were used to running before, even when you're jogging, because that is our tradition. So thank you so much and welcome so much for coming. We hope you have enjoyed the hospitality, the culture, and the academic environment at the University of Eldoret and in Kenya, and Kenya in general. Now to the organizing committee, the volunteers and the support staff, please also accept our sincere appreciation for the excellent coordination and dedication that ensured the smooth running of the symposium. Normally it takes a lot. I know that since this organization started, I think it started when there were some turbulence in higher education within this country, and with the changes in the programs and sometimes having very short times to dedicate to things like planning of symposia like this. But I think you have already, you have overcome that and at least came up with a plan which has resulted into this symposium taking place up to where now we are ending it. Now, as we come to the close of this symposium, we hope that the deliberations and the networks built here will continue to inspire impactful research and strengthen Africa's contribution to global scientific advancement in genomics and molecular biology. I therefore take this opportunity on my behalf, on behalf of our Vice Chancellor, and also on behalf of the entire University of Eldoret fraternity, to wish you a safe journey back to your respective destinations, and also to wish you every success in your future research endeavors as we continue to see how we can solve the problems and aspirations of our different communities that we are serving through innovative research and coming up with what will be relevant now and what may be relevant in the future. As a country, we are in the transition of offering our curriculum, and I'm sure most of you might have heard of that. Now, all those will require the participation of our partners, colleagues, our international friends, because we are going to move from the traditional teaching to more of an innovative way of teaching and allowing our students to explore more. So those who are already on it, and even the students who are on the research right now, that is where we are going, and we want to encourage you. So with those many remarks, ladies and gentlemen, allow me to thank you once more and to officially close this symposium. Thank you so much.
Thank you, Prof. Raburu. So let's applaud him once again. Yeah, thank you for closing the function. So we move to the next section. Yeah, but before we begin the final details of that next section, which is focusing on keynote speakers. Yeah, Prof, we had 8 keynote speakers. Yeah, talking on different topics. And some are here, some have left, some presented online. Yeah, so may I just request keynote speakers who are around to stand. Yeah, so these ones are still around. Professor Cyprian Katongo is from the University of Zambia. Professor Paul Grobler is from the University of the Free State in South Africa. And Professor Ralph Tiedemann is from the University of Potsdam in Germany. Yeah. Thank you. You can take a seat. So 5 of them, 3 presented online and 2 have left. Yeah, but we have some certificates here for keynote speakers. So Prof, once again, we request that you come in front so that you give these certificates of participation. Yeah, so we have Professor Ralph Tiedemann. We have Professor Paul Grobler. We have Professor Cyprian Katongo. Dr. Rispa Tororei, I think she has just left. Then Josiah Fuja left in the morning, presented and left in the morning because of this busy schedule. Yeah, then we have Professor Lilian Kinywa. She was here in the morning, then left for Senate.
I left her where I was.
Yeah, so, okay. Yeah. Then we have Professor Raphael Mrode. He presented online. He's a professor in the UK. Then Professor Anise Happy is a professor in Nigeria. She also presented online. So the next section is the planning committee. Planning committee members. If you are a member of the planning committee, please just stand. Yeah, so that the DVC can see you. Yeah, so we have Vivian Tuwei, who was in our place. We have Dr. Kenneth Bukania, who's a member. Then we have Dr. Emily Chemoywa. Yeah, then we also had Samuel Kariuki, who has left. Including myself and Director Research, who is away on official duty. So we also just want you to present this to them. Yeah, so, yeah, Emily, you are the first one, seemingly. Yeah.
Yeah, she likes sitting on my left side, so. Okay.
Yeah, Vivian, yeah. Vivian? Okay. Yeah.
This one means you're supposed to come back.
That is who? Benson. Benson Nyongesa didn't manage to come. Kennedy, Dr. Kennedy Pukania. Yeah, then there is James Barasa, who is myself. So the next group is for delegates. And we had delegates from different parts of Kenya and also different parts of Africa. So we will just be calling, then you see them as they come to receive their certificates. Anthony Mabere did not manage to come, presented online. Amadine Kunil presented online. She didn't manage to come because of the crisis in the Middle East. Joan Kiplagat, are you around? Joan? Yeah, then Professor Hans Michael Latov, yeah, he presented and left this morning, yeah, back home. Then Blasio, you are the next. Blasio Bisereka is from Uganda, yeah, Busitema University, Uganda. Ezra Kirarei, next. Ezra Kirarei. Next is Hilda Chepleting. Next is Sarah Waswa. Next is Feng Cheng. I think Ralph will receive for him. Yeah, just a minute. Just come. Feng Cheng presented online, but is a postdoc fellow in Ralph's lab. Yeah, so he will receive and take it to him. Linda Sitati presented online. Huma Hills is a Malawian. He presented online. Yeah, Sitati Catherine Nasambu. Catherine is here. Fourth year, biological sciences. Catherine Sitati, Anne Laigong, from Cambridge Graduate School, Olivia Dimoff, she's left, okay. Edith Kurui, yeah, she presented online. Salina Chepto, Rono. Vivian Chilumo presented online. Bashir Ali is a Nigerian, presented online. Okay. Bashir Meriko is Malawian, presented online. Sharon Auma. Sylvia Chepkosgei. Sylvia. Sulaminte is Malawian, she presented online. Daisy Naflasmiu. Daisy. Next. Naman Ondego Arodi. Naman. Olivier Dayo. Olivier Dayo is from Benin, but is currently studying in Germany, but he has also studied in Kenya previously, at Kenyatta University. Next. Purity Muhongo. Purity. Then Sulukalaini Shehu is Nigerian, he presented online. Okay. Emily Chemoywa. Emily. Oh, presentation also. Yeah, then James Barasa. Yeah, so the next session, as we wind up, it's another important session. So our guests, we have something small for you here, for you to go with back home. So, Professor Cyprian Katongo, just come and take something small. It's not much, but as you go home, you will be looking at it.
Thank you. Thank you very much.
Professor Paul Grobler, there's something small also for you to take to your Bloemfontein. Professor Ralph Tedman, there's also something small for you. They should have more than this.
Then, yeah, Professor Miriam Kinywa. This is hers. Dr. Rispa Tororei. She has just left. No, more is coming. Some more is coming. Yeah, so this one's are done. Yeah, so for Professor Anissa Harper, this is hers. Then there is a Professor Raphael Morode, this is his. We will see how it will be taken to him. Then, of course, the DVC for coming. Yeah, there is also something for you. So I request Salina Rono to come forward and present this to the DVC. Yeah, just come. Yeah. Yes. Yeah, this one.
More blessed to give
This
than
one.
to receive.
Yeah, so thank you, thank you. Yeah, so for the guests who are away, we will see how these certificates and their gifts will reach them. Yeah, thank you so much. Yeah, on this note, there isn't much for me to add, so we just come to the end of the session. So may I request one person to close the session with a word of prayer. Then we will arrange. Yeah, we will arrange. Who can close with a word of prayer? Sarah? Sarah, are you able to close? But as she prepares to close with a word of prayer, yeah, we will have some interaction out there to relax. Yeah, so there is tea after this. We take tea and then relax a bit. Then by 7 or shortly before 7, we will arrange for taxi to Eka Hotel, where we will have our dinner. And dinner is there for everyone. Yeah, everyone. So don't leave before that. Thank you. Sarah? Or Daisy?
I think we should stand up, be on our feet as we pray. Okay, let's believe and pray. Our Heavenly Father, we want to say thank you for your mercies and your grace, Lord. We want to say thank you, my Father, for being with us, Lord. As we started this symposium, Lord, we asked for your presence, Father, and we want to say thank you for being with each and every one of us, my Father. We want to thank you for the journey mercies you've granted us for the 3 days that we've been here, Lord. And as we close up, Lord, we want to ask you that, Lord, you're going to continue being with us, my Father. Wherever we will be traveling, my Father, we want to ask you that, Lord, you're going to go ahead of us, my Father. As we continue with our various researches, my Father, in various fields, oh Lord, we want to ask you that, Lord, again, you're going to be with us. You're going to bless us, and you're going to open our minds and our inner understanding, oh Lord. We bless you, we'll worship you, for we know that there is none like you. In Jesus' name we do pray and believe.
So as we leave, let's take a group photo out there, I think, before we take tea. We go for a group photo, then we take tea.